Good afternoon. I'm Ruth Lehman. I'm the director of the Whitehead Institute, and I want to welcome you all to the Symposium, AI Advancing Foundational Biology. Thank you for being here. This room holds biologists, computer scientists, engineers, clinicians, and founders, and that's an important mix. The discoveries we will hear about today happened because of the diversity of our experience and the shared passion for curiosity and innovation. We launched the Whitehead Innovation Initiative 3 years ago to help biologists and computational scientists to work together, not in parallel, but in true partnership. Artificial intelligence is changing how we ask biological questions, not just how we answer them. That shift requires new collaborations, new tools, and new ways of training the next generation of scientists. In the last year, we have supported 11 labs here at Whitehead to build shared platforms now used around the world and trained the next generation of scientists who move fluently between computation and experiment. The symposium brings together researchers who are shaping that future. And you will hear from leaders across the field, people building foundation models for microbial genomes, mapping protein interactions at proteome scale, decoding gene regulatory mechanisms in cancer, and designing systems to predict cellular function. And I'm really grateful for those visitors. Some of this work is happening here at Whitehead, and of course, a lot of it is happening elsewhere. All of it turns to a common horizon, biology that moves faster, asks more and possibly sharper questions, and opens doors we didn't know were there before. Of course, for us, the symposium and our activities around AI would not have taken off as they did, wouldn't it be for the support of Michael and Victoria Chambers. Their vision and generosity launched the Innovation Initiative and has given our researchers the freedom to take on risks, build platforms, train students who will carry this work forward for decades. So I really thank all of you for being here. I thank our speakers for coming to Whitehead today. And now I would like to turn over to Matteo. Matteo Di Bernardo is a graduate student in Ian Cheeseman's lab, and he is really the architect of today's program, and he will walk you through what to expect next.
Thank you, Ruth. As Ruth said, my name is Matteo, and I'm a computational biology student in the Cheeseman Lab. Like many of you, I've watched artificial intelligence go from a specialized tool to kind of a broadly accessible resource that allows us to change how we design experiments, analyze data, and think about biological programs. The Whitehead Innovation Initiative was created to answer this challenge and how to integrate these tools into the work that we do here at the Whitehead. And over the past year, we've expanded what the Innovation Initiative has done, bringing to the Whitehead leading researchers like James Zhao and Marinka Zitnik. And we've really tried to facilitate both these seminars, but then also internal talks and primer series to empower the Whitehead community to pitch bio-AI projects, as well as dinners that people can use to interact with people across Boston that are working in this area. Today we're convening for our marquee event of the year, which is our symposium to understand how AI can advance foundational biology. The scientists you'll hear from this afternoon are biologists first, asking the questions that we care about here at the Whitehead across gene regulation, cancer progression, protein organization, and microbial evolution. They're building models and really trying to explore these subjects in a deeper way, and I'm sure that we'll all have the opportunity to learn from them today. Now let me walk you briefly through the afternoon. We'll start with 5 talks from researchers both at the Whitehead and beyond using machine learning and computational methods. Each talk will be followed by a short Q&A. We'll try and keep things to 5 questions. I'm just letting you know in advance, just so we can keep the program running. But we will have a break and a poster session following the event, such that you can ask the speakers more questions. The final part of the talk, or of today, will be an exchange where Ruth will sit down with all of the speakers and we'll have a conversation about what we've heard here today and kind of explore some of these topics and the horizon of AI and biology further. And as I said, at 5:30, we'll open the doors for a reception and poster session. You'll have the chance to interact and hear about the 11 projects that Ruth mentioned that have been funded through the Innovation Initiative and speak to trainees that are working in this area. And with that, let's get started. So please join me in welcoming our first speaker, Dr. Christina Leslie from Memorial Sloan Kettering Cancer Center. Thank you.
Thank you for the invitation. It's an honor to be here and be part of this event. So I changed my title slightly. I thought I might have time to show you 2 different models, but I'm definitely going to talk about neural ODEs. All right, my lab at Memorial Sloan Kettering, what we do is we use single-cell, spatial, and 3D genomics to try to understand the regulation of gene expression, but also how cells interact with each other in tissues and tumors. These days, we're doing a lot of AI models for regulatory genomics, but with wonderful collaborators, we also work in basic and cancer immunology and cancer epigenetics. So while mostly I'm going to show you the AI work, you'll see peeks into that other world in some of the problems. All right, I'm going to start with a disclaimer, which is advancing foundational biology sounds like we are going to talk about foundation models for biology, but I'm actually not going to talk about foundation models. We do in regulatory genomics. We're at an interesting time since there's been 10 years of AI in regulatory genomics, and Alpha Genome was just published in January. And we have what could be considered a foundation model, taking a megabase of sequence from the reference genome and predicting thousands of epigenomic and transcriptomic tracks, okay? And this is useful for variant effect prediction, but there's still unsolved challenges. So this is from their own discussion, capturing the influence of distal regulators, regulatory elements that are more than 100 kb from the promoter, still a challenge, even though we have this big 1 megabase context. And also recapitulating tissue-specific patterns across cellular contexts. The models don't actually generalize to new cell types, these ones, okay? So, there's a citation at the bottom. Some of my students and I did a short comment on the state of predicting the regulatory genome, if you want to take a look. Okay, what am I going to talk about? I'm going to talk about using AI models with the right data and in the right biological context to actually learn something new, all right? And for us, we've become very excited about single-cell chromatin accessibility, right? So we can map with a single assay accessible elements in the genome that it's cell type specific. It's where transcription factors are occupying the chromatin, and we can do it in single cells. And we can measure single-cell chromatin accessibility and single-cell transcriptomics at the same time, in the same nuclei. So that's empowering. And we also have very good quality information about this dynamic 3D organization, interactions between distal enhancers and promoter elements through 3D genomics, high-C, high-CAR, 4C, high-CHIP. So what I'm going to tell you about today, I'm going to give you 2 vignettes. First, how we can exploit 3D genomics to go far away in the genome and learn powerful models of gene regulation. And then second, I'm going to show how we can exploit single-cell chromatin accessibility, single-cell multiome data to learn functional models of the dynamics of cells. Okay, and I have too many slides, so I'm going to go fast. All right, so first vignette. Again, interesting biological context and collecting the right data. So this is a collaboration with Danwei Huangfu and Effie Apostolou. Danwei Huangfu is an expert in pancreatic differentiation, has a beautiful system of guided differentiation of human ES cells towards beta cells. And what we've done at every stage of the system, the Huangfu lab has generated single-cell multiome data, as well as 3D genomics data, high-CAR, okay? And on the right, what you can see is at different stages of pancreatic differentiation, human ES cells, definitive endoderm, pancreatic progenitor 1, you can see enhancer-promoter interactions being set up over an important gene, PDX1. It's a pancreatic transcription factor, important developmental gene, and a diabetes gene. And so we can try to use this data to model gene regulation, understand enhancer rewiring in differentiation, and also understand non-coding genetic variation associated with disease, associated with diabetes. Okay? All right, so I just want to show a little bit of the single-cell data. We're going to go to pseudobulk in a bit, but it's actually very beautiful data. I'm showing a UMAP of RNA and the single-cell attack, the chromatin accessibility separately. And what you can see is that at later stages of this protocol, as you start to have early and late endoderm endocrine cells develop, there is more structure. And in particular, in the chromatin accessibility, we can see this early, actually at the PP2 stage, these neurog-G positive cells that seem to be giving rise to all the later endocrine cells. And this embedding of single-cell attack, it's actually a method from our group where it's a sequence, DNA sequence-informed embedding, where we're embedding DNA kamers and cells into the same latent space and allowing them to pull each other around so that we get this, these developmental trajectories. Okay, so now we're going to go to a pseudobulk level, different cell types, and we want to use 3D genomics data, okay? How do we incorporate 3D connectivity like Hi-C or Hi-CAR into models of gene regulation? So we went back to a model we published a few years ago called GraphReg, okay? And this is actually still the biggest context model that there is out there. It's 6 megabase of sequence context. And the idea is that we're going to define a graph from Hi-C edges on the genome and pass information across these graph edges, okay? So you can think of the genome as nodes with graph edges between them if there's a 3D interaction. So what the model looks like, you have this big one-hot encoded 6 megabase of DNA sequence. The first path down the model, you have convolutional layers and then dilated convolutional layers to predict 1D epigenomic data. And then you pass the information, this kind of motif-like information, to a graph attention network and pass information across Hi-C edges, and we predict cage-seq. We predict gene expression along the genome, okay? And we can use feature attribution to figure out which distal enhancers are important for predicting the expression of which transcription start sites. Okay, what do we have to do here? We have 3D genomics data through Hi-CAR. We have pseudobulk multiome, but we don't have everything that we needed from before. So the single-cell RNA readout is a 3' capture, okay? We don't have something like transcription start site activity, so we want to supervise at the gene expression level. ATAC-seq alone isn't that much information. Okay, before we had multiple epigenomic tracks. So what we do is train a model to go from DNA sequence to ATAC, and then we use a representation from that sequence model together with the ATAC itself to represent our input. Okay, so what we do putting it all together is we have a hybrid graph, okay? So bins along the genome, interactions between bins represented by high C. And then also for every gene, we connect it to any bin that contains a known transcription start site. And then we supervise at the gene expression level, okay? So now we can predict gene expression from sequence and ATAC informed by 3D connectivity. All right, so this is an example of the feature attribution from the model. Okay, so we're looking at the gene 1cut1, and there's a model at every stage. I'm showing in loops the high CAR, so the connectivity. And then there's a red track, which is the feature attribution. And if I had a pointer, I would point, but you can see in boxes that there is an enhancer identified by feature attribution in the model that is actually more than 600 kb away from the transcription start site. It also contains a common variant associated with type 2 diabetes, and later we found there's a rare variant associated with monogenic diabetes. And this enhancer, very far away, was validated by the Huang Fu lab in this recent publication. Okay, so it really does regulate this gene in a stage-specific manner and can explain dysregulation of gene expression associated with disease. Okay, last slide on this is just if you do this systematically and you look at all fine-mapped common variants associated with type 2 diabetes, filter for score, require a significant 3D interaction, you can actually find hundreds of variant gene links that are more than 500 kb in genomic distance. Okay, so perhaps there's a lot of distal regulation that we're missing that can explain common disease, also like monogenic disease, by looking at the right data in the right system. Okay, so now I want to switch gears and talk about what my title originally was, which is using neural ODEs to learn dynamics from multiome data. And people are probably familiar with trajectory methods and pseudotime for single-cell RNA. There are many of these methods, right? So the basic idea is that we have a destructive technology. We don't have real-time course for trying to infer a pseudotime that some cells in our single-time snapshot may be developmentally earlier than others, okay? And we infer these trajectories. And the question is, can we learn a functional form of these dynamics, okay? And we were inspired by RNA velocity, that might be familiar to some people, where the authors of RNA velocity tried to write down an ordinary differential equation relating unspliced and spliced reads, and putting a phase diagram, the idea that when you turn on a gene, you see the unspliced product before the spliced product. You know, so there's some precedent for using ODEs, ordinary differential equations, but this method also has its drawbacks and lack of robustness. You can only do the ODE for some genes, the velocity genes. It's also multi-step, okay? It's not a single model. You have a model for every gene and then infer these kind of latent times, okay? So what we're going to do is a single model that we learned from the data. It's called Dynavello. And the important thing is we're not just using gene expression and inferring RNA velocities, but we're also using transcription factor motif accessibility, sort of motif activity as a trans state of the cell, okay? And the idea is if you have a motif for a transcription factor, you can get a single cell accessibility, from a peak atlas, and we use ChromeVar, okay? So a kind of summary of the activity of a transcription factor, and we use all the transcription factors that are expressed, okay? And we are going to learn a functional form of the RNA and motif velocity fields and latent times for all the cells. Okay, I think I'm probably going way over time. So here's the basic idea of the model. So we have gene expression and motif accessibility for every cell. We embed, we encode into a latent space and then decode back into the original space. We learn the dynamics in the latent space, the lower dimensional space. And what we're learning for every cell, we're learning its starting point, its initial time zero in the latent space, and its latent time. And then and we're learning a functional form of the dynamics of the vector field encoded by a neural network. Okay, we use RNA velocity to.
Do you need laser?
It's okay. It's okay. It's okay.
This is active.
Okay.
Just for the lasers.
Just for the laser. Okay, we do have a laser. Okay, I will try to dual hand it. Okay, without. Okay. We're going to use RNA velocity for the velocity genes to supervise. It's in the loss function, so it gives us a little bit of help on direction. Okay, so neural ODE, we thought there's so many of these trajectory papers, we need to do something hard, right? And we decided, also because we have wonderful collaborators, to look at the dynamics of B cells in the germinal center reaction, okay? And as you know, this is part of the how B cells mature their antigen receptors. They proliferate in the dark zone and undergo somatic hypermutation, go to the light zone, either get T cell help or not. They might recycle back, and there's multiple exits, either as pre-memory B cells or plasmablasts. So very, very complicated dynamics, and everything is governed by transcription factors. So it's a good place to use this model. Okay. Right. So here's Dynavello applied to germinal center B cells. This is data from Ari Melnick. This is wild type. These are in vivo from mice, like in vivo dynamics. And what you can see in green are the dark zone centroblasts. In blue are the centrocytes. And I think we're nicely capturing this basic bifurcation of, do you proliferate in the dark zone or do you transition to the light zone? And you can even see this kind of recycling back into the dark zone and the multiple exits, okay? And just RNA velocity itself, if you look at the latent times, I think you can see it's not capturing this bifurcation. Everything is just going towards the exits, okay? So, so this is reasonable. We can, this one. Okay, we can also, and many models can do this, we can organize cells by latent time, and we can start looking at the inferred motif accessibility and gene expression along latent time. And I know this is small, but what I want to emphasize, so transcription factor motif accessibility is kind of activity is in green and pink, and the expression is in blue. And you can see the dynamics of transcription factor activity are much stronger than the RNA expression dynamics, okay? And it's pointing to important transcription factors that might suggest drivers like SPI1, SPI-B, and of kappa B1 and 2 for pre-memory commitment. Okay, we have mutants. Okay, some of these mutations like ARID1B, heterozygous loss, you see in B-cell lymphomas. What do they do? We can run the model on this data too. Phenotypically, what happens is you lose recycling and you exit as a kind of immature pre-memory B cell. So it's no recycling, bigger dark zone, and exit from the light zone. And you can start seeing there's certain transcription factors that behave differently in the loss of function mutant versus wild type. Okay, so like stronger IR-8 activity. Okay, so far this is very close to the data and just organizing by latent time, but we can do more because we have a functional form of the dynamics. So we could do Jacobian analysis, and we can ask if we change gene expression of certain genes by a bit, what RNA velocities change. Or we can do an in silico perturbation, especially for a transcription factor. We can set its expression and activity to a minimum value through the whole system and see what velocities change. Okay, and that's what we're doing here. We're inferring regulatory edges from either Jacobian analysis or in silico perturbation. You know, perturbing one affects the other. And we can also see that these edges are dynamic, right? So it's not like a regulatory network is just a diagram, it's changing through the system. Okay, and once we do this, we can kind of put it all together and give the cartoon version that you see in reviews, and you can get about everything that is known about the germinal center reaction and transcriptional control, and you can also predict certain edges that are lost in heterozygous loss of ARID1A or CTCF. Okay, 2 more slides. 3, actually. Okay, so I, very quickly, right, and there's a bioarchive preprint if this is all going too quickly. We had the wild type data. Could we predict from the model what happens in a loss of function mutant? Could we predict what happens when you lose ARID1A? So to some extent we can predict the major effects. So what we can do is do an in silico perturbation setting ARID1A to its minimum effect. And you know, I don't actually like these visualizations. This is like the change in velocity. And you might sort of see, oh, like maybe we're losing sort of or increasing exit and losing recycling. But more systematically, we can check whether we can look at genes that significantly change their velocities and make those predictions. And for example, we can predict this higher activity of IR-8. Okay, if you have a mutant, could you predict perturbations that rescue the loss of function phenotype? Okay, so same idea. We just try all in silico knockouts of transcription factors, and then we see if the predicted change in velocity pushes genes and transcription factors in a direction consistent with the wild type. You know, are we rescuing? And you could say, you know, like we're looking at perturbations that rescue RNA velocities or rescue TF velocities. And here's a predicted one, tox knockout and ARID1A is predicted. Okay, so, and of course, you know, can we validate any of these predictions? And so we have validations for 2. Why these? Why not tox are the really strong ones? You need a mouse, right? So and we're doing this in revision, so we had to have the mouse. But for example, what we can see, at least phenotypically, is this, you know, BCL6 rescue perturbation. Here's the single knockout of ARID1A, single knockout heterozygous loss of BCL6, and the double knockout. And it is rescuing this dark zone phenotype. of the ARID1A loss. And similarly, I'm not showing the data, but there's a whole paper that will eventually come out from our collaborators that CTCF heterozygous loss rescues ARID1A loss. Okay, and more to come in terms of validation. Okay, that's it. I want to thank all the amazing postdocs and students who did the work, and I'm happy to take questions if I have any time left.
Questions?
Thank you for your wonderful talk. So could you please comment on the future development of the AI model using partial differential equation to incorporate spatial multi-omics profiling so that we can have cell-cell communications and cytokine signalings incorporated into these predictions?
Yeah, so that's a great question. A number of us are trying to look at spatial transcriptomics and model systems. I think we're, what we can perhaps do, not with partial differential equations, but just with this kind of approach, is look at pairwise interactions that influence each other and sort of say that it's a dynamic process. So we're working on that. Other people are trying to look at developmental spatial data and derive neural OD methods. And there are some extensions. Partial differential, like there are a lot of formalisms. There's flow matching, there's optimal transport, there's this kind of neural ODE. I think it's starting, right? We don't have the ideal data for it, but it's absolutely, people are looking at it.
So within the last part of your talk on the ODEs, you talked about transcription factors or motifs, and you even showed some results for how they are able to predict the velocity of a gene expression. But I'm just wondering if in the latent representation you have a finer level of granularity that might represent the actual sites of open chromatin, and can you then identify the relative importance of distal enhancers versus
yeah, so that's a great question. So this is not as mechanistic as it could be, right? We're not modeling individual sites. And there's just reasons for sparsity and high dimensionality, right? So we're summarizing very high dimensional but sparse single cell attack data by a bunch of transcription factor motif accessibilities. That's very interpretable. So we have features that are interpretable, right? And lead us directly to what do we perturb, experimentally. But it's not the same as representing individual enhancers. That could come by taking parts of the first part of my talk and combining it with the second, but future work.
Any other questions? Okay, I think we're going to thank Christina, and next up we'll have Hani from UCSF. Thank you.
Hopefully someone will turn it on. I'm up there, or is it on now? Yes or no? Okay, perfect. Awesome. As Matsuo mentioned, I'm Hani. I hail from UCSF, where I'm associate professor. I'm also a core investigator at ARC Institute and an AI research lead in the Computational Technology Center at ARC. So today I'm going to cover a bit of work from my own lab, but also some of the work that we have been doing as part of ARC and as part of our institute initiatives, specifically the Virtual Cell Initiative. Okay, so this is my disclaimer slide. I've bolded Tahoe Therapeutics because I'm going to cover some of the work that I've been doing with that company as well. So just bear this in mind. So when I started my lab 10 years ago, one of the first questions that I started with, kind of in collaboration with one of my mentors, Kevin Shokat at the time, was fundamentally this question of why drugs fail. Like 90% of drugs fail. We've all heard of this. About half of them are failures due to efficacy. And so what is the fundamental reason for that? And during our conversations, Kevin and I kind of landed on this idea that this is our current shape of how we do drug discovery in oncology. So you have a heterogeneous population of patients. You find a specific drug that represents this population. You isolate that target, either via really biochemical assays or cell-based assays, and you do your drug discovery in that simplified system. You also do your SAR, structure activity relationships are mapped out, and it's really the final few handful of top candidates move into more sophisticated models, where in oncology is basically xenograft models, patient-derived xenograft models, or cell line-derived xenograft models. So looking at this picture, there were two immediate parts that kind of caught our attention. One is that much of your drug discovery or drug screening is really happening in the context of this very simplified model system. So in machine learning, we are all very familiar with this concept of like overfitting to these kind of simpler environments. So the more biological, the more complex biological system is missing. And also, really throughout this process, even late in the xenograft models, the true heterogeneity of the patient population is not really captured. So we set out to solve this problem. It was really a happy coincidence that we could solve it via this platform that we worked on for many years called Geneva. And it was really a confluence of a lot of technologies that we borrowed from our colleagues at the time. Jonathan Weissman's work on tumor single-cell profiling, sample hashing, Perturb-seq, Jimmy Yee's work on a SNP-based deconvolution, and a lot of other single-cell technologies. And we kind of put all of this together in our lab to create Geneva as a model that brings, as a platform that brings cell village concepts to tumor models. So basically, what you do is you take tumor models, for example, cell lines or patient-derived organoids or xenografts, and you mix them together into one unit of observation, and you either implant these in mice or you have them in culture. And this model then receives a perturbation, usually a drug, and you do sample hashing and single sequence everything. So what comes out of the assay is fundamentally a transcriptomic assay, but as a single cell resolution, we have shown that it learns and it demonstrates mechanism of action, mechanism of tolerance, just tells you a lot about what a given drug does on a very heterogeneous number of tumor models kind of simultaneously, and it scales across that dimension quite effectively. And this is really the first experiments that we did, and I really like to show this partly because of that. And now this is a heterogeneous population that we have built. Again, it's kind of a basket trial type mechanism. This is just a 2D culture, not tumors in mice. And we have treated these cell line models with two drugs, ARS-1620, which is a KRAS inhibitor, and vemurafenib, which is a BRAF inhibitor, V600E mutant specifically. And you can see what the data looks like. You can easily distinguish the cell line populations, and you can label them. Again, this is based on their individual SNPs, but you can really also go off of the gene expression in a plot like this. You can also do sample to convolution, so you know which drug each of the cell lines we're seeing. And I'm pointing, and there's a lot of data in there, and you can go deep dive into it, but I'm just really pointing out A375 as an example of a cell line that's very sensitive to vemurafenib, and you can see there are almost no green cells. So that's basically, you get both the gene expression responses, but also these cellular phenotypes as well that I'll go back to. Now, this was cell line based, as I mentioned. You can really even do this in the context of more complex tumor models as well, and that was more important to us. So you can actually create these complex tumors in mice, heterogeneous tumors in mice, and the mouse receives the drug. And again, showing KRAS inhibitors as an example, you get a very similar picture. You have all these different cell line xenograft models at this point, and you can actually see which are the cells that are responding to mice being treated with a KRAS inhibitor, in this case, ARS-1620, which specifically targets G12C, and you see the lines that have G12C mutations in them are the ones that are disappearing from the population in relative terms. So what is nice about Geneva is that we could also do it in vitro and in vivo simultaneously. I gave you this context that we think in vivo complexity matters, and intuitively we are believing that, but now we actually had the data to answer the question of actually does it contribute anything? It turns out it does, as expected. There are many cellular programs that are shared between what you see in vitro and in vivo, but there are also quite a few that are exclusively seen in vivo, or at least a lot more pronounced in vivo. And there's one example of this that I'm showing here on this slide. So the two axes are in the in vivo and in vitro of the same exact, basically, experiment. So this paper actually came out just recently. The work was done 4 years ago, but you know how it is. So a big shout out to Johnny, who was the lead graduate student on this work, and also another graduate student, Timmy. So feel free to do a deeper dive on Geneva. You can find it on Nature Cancer website as we speak. Now, we thought that, again, our premise was that if we move our drug screening into these kind of more complex models, taking advantage of platforms like Geneva, we should be able to find drugs that are more likely to be efficacious ultimately in patients. But the reality is that you can treat models in mice or whatever sophisticated model you can find, but regardless, it's still a model. So humans are different. The immune system is different, the metabolism is different, all these components. So this is not going to take you all the way, right? So that is basically a key limitation that no level of experimental modeling in simplified or even complex system is going to overcome. So this is basically has been our kind of guiding principle for our kind of in silico screening approaches. The idea being that when you look at kind of a trial, a clinical trial, you get a picture like this. This is the heterogeneity that you would see when you run a trial. And this is, again, a very successful trial. These are all G12C patients. Their tumors have G12C mutations. They're getting a selective drug that you can even see, because it's a covalent drug, you can actually see it binding to the target. If you run a gel, you will see it shift. And still, you see a broad range of responses, all the way from very good response, strong response, to all the way to the tumor not caring at all, right? So what really motivates this in humans, and how can you actually be able to replicate this, right? How can we take data, all the data that we have from in vitro, in vivo, all kinds of experimental models that we have, learn from that data, but then be able to predict what happens in a cell, cancer cell, in a patient, right? Because we can collect data from that patient tumor, but we cannot do experiments on the patient, right? So we want to do that experiment in silico. And this is a problem that we call context generalization. How can we have this universe of data that we train on, but then the new context comes in, which is the patient, and we want to predict all these drugs that we have here, how would they interact with this tumor in this patient in this state that the tumor resides in. So this is basically kind of a manifestation of virtual cells. So I mean, a lot of you have heard about virtual cells kind of emerging as kind of a holy grail in AI modeling these days. Again, it's a very mushy concept, as many of you will come to realize if you haven't. But the reality is that to me, it's kind of a banner that we're all fighting under. So the bottom line is, from my perspective, this is the definition of it: how to do context generalization to a new cell in a given context that we cannot do experiments in. So be able to do that experimentation in silico. And in the context of oncology, for example, ultimately, the goal would be to really faithfully recapitulate and model what happens in a tumor inside a body, kind of all the systemic responses and how they impact the tumor's progression. But in reality, the bar to meet is really the xenograft models that are the tools that everyone uses for drug discovery. So if we pass that bar, I think we are better off, basically. So even before we get to that ultimate success rates, there are wins that we can try and meet. Okay, so what does it take to build a virtual cell the way that I have described it? Again, the idea, what makes a virtual cell is that we want it to be able to generalize beyond its training universe, right? So the difference between kind of a foundation model and a normal predictive model is fundamentally that I can, if I'm like in a cell line universe or a PDX universe, I can do predictions in that context, and that's fine. But here, it's one step more challenging. We want to train in this universe, but then be able to generalize to a new data universe that's slightly out of this distribution from our original data set. So that's like one principle that we have to always remember. The premise that that is guiding the way is that ultimately gene regulatory networks are shared. That's what shapes every cell in our body, and it has, like, while it's complex, it's like bounded complexity, like the rest of biology. Biology is not chaotic. It's complex, but there is bounded complexity that we should be able to ultimately, given enough data and enough parameters in our models, be able to abstract. Now, over the past few years, and almost a decade now, there are also lessons to be learned from language, from protein, where the successes have been, where we can actually see what has worked and what hasn't, and maybe get some ideas from that. Okay. Stop moving? Okay. Yeah. Awesome. So now in biology, of course, we always care about function. Cell biology is no difference. Oh, wow. Lost it? Not super responsive. So again, we care about cellular function. In general, in biology, we care about function. Function is hard to measure scalably. So we look for proxies, and we model those proxies. For example, we have all been fascinated by protein structure modeling because in proteins, there is a tight structure-function relationship. Right? In DNA, no one does a structure modeling because there is no function relationship in DNA. So we do sequence to function relationship most of the time. So when it comes to cells, what is that proxy, right? Really, thanks to advances to single-cell genomics, we have come to realize that there is a cell-state function relationship in cells. So if two cells reside in the same state, they probably do the same function. And how do we define state? State is basically a lower projection of a multi-omic measurement that you all have come to see and love in the form of UMAPs. Right? So that's basically what we are trying to model. We are trying to model cell state, but in the end, that's a proxy for cellular function. Now, for proteins, we had this really privilege of, again, everyone thinks that PDB was very important, and it was very important, but also equally important was really the wealth of all the coding sequences that we had access to, and all the evolutionary patterns we could see. All the changes that evolution had tolerated could actually tell us something about, again, sequence structure relationships in proteins. In cells, we do not have that because, well, we haven't measured cell states across tree of evolution, right? We have like some humans and mouse, and that's about it, right? So while evolution is exploring all these different sequence variations, we don't really have that. So evolution is doing these perturbation experiments that we do not have equivalent of it in our large databases, right? So the manifestation of it for kind of cell-based modeling becomes perturb-seq, right? So it is the kind of system where you do your perturbation yourself and you measure the consequences of it. So that's why perturbation is really important for us, because it proves causation. It really makes the data much, much richer when it comes to modeling. Now, one of the ways that we have done this is by extending Geneva. So Johnny and an old friend of mine, Nima, joined forces and started this company called Tahoe that has taken Geneva and made it a lot more scalable. And a big output of that was this data set called Tahoe 100M, which remains the largest single-cell perturbation atlas, which is about 1,100 drug dose combinations measured on about 50 cell lines. And these cell lines, it's a basket cell line. So we pick them for diversity, all kinds of drivers and all kinds of mutations. And the drugs were also themselves quite heterogeneous as well. And this is like what the data really looks like. So it's, again, a massive data set. What I'm showing you here is, I think it's a 1 over 1,000 subsampling of that data set. But again, it's very minimal batch effects, very beautiful data. If anyone who hasn't used it, you should. And what comes out of it is not only gene expression changes, but all the type of cellular and molecular kind of phenotypes that we have been able to build into Geneva over the years. That includes the cell counts themselves, cell cycle phasing, the distributional changes in gene expression landscape, and so on and so forth. And you can do this to, for example, measure the survival level changes in responses of the cells to different drugs. And this is one example, right? Again, I keep going back to G12C because I've had a lot of fun with this drug over the years. But again, you can see if you look at this drug and who is responding to it in terms of cell counts in the population, what comes out of it is KRAS G12C as being the most important determinant across the 50 cell lines, right? So you can understand the mechanism of actions of drugs. Going to the molecular gene expression levels themselves, you can create these molecular phenotypes, which, as you can see here, the cells respond in coherent ways, the ones that are sensitive. And you can actually use this information to classify drugs as well, or even find the ones that are potentially misclassified. So this is kind of how well we can predict the mechanism of action of each drug in the data set based on these molecular, quote-unquote, molecular phenotypes and signatures. So we have taken all this perturbation data, mixed it with all the data that's out there. We actually went through this process of collating all public data sets as part of our SC base count effort, which was one of the first agentic applications of AI to biology that I know of. So we put these data sets together as part of what we call ARC Virtual Cell Atlas. So Tahoe contributed Tahoe 100M, we contributed SC base count. And the idea is that the two of them, they give you a kind of training data for AI-ready applications, basically. Now, depending on how much, I have no sense of time. Maybe tell me how much time I have. But I'm going to tell you a little bit about now the work that we have done on the modeling side of this data. And I'm going to talk about it, but as you can see, there's a large team at ARC basically dedicated to solving this fundamental problem, which is, again, perturbation modeling. So how do we define a problem? The problem is this. We are starting from a population of cells, right? And we want to figure out where does this population end when after perturbation, right? So what we want to model is that outcome. So that outcome is influenced by the perturbation and also the heterogeneity of the original population plus some noise, right? So this is the generative process that we are trying to basically model. So how do we do this? So our solution, which is already available as a preprint and hopefully in print soon, is called state. It's a two-part model, as I'll tell you. One of the key modules of a state is a state transition, which is a model that actually understands this perturbation transition, that the response to perturbation, basically. So how does, what is unique about this model? It's actually a set transformer model. So what you are modeling is not one cell at a time. It's basically populations of cells. And these populations are covariate adjusted, right? So you're basically taking a few cells that are supposed to be, as far as we know, experimentally should be the same, right? They have been in the same condition, have seen the same environment, all of that, right? So that's what I mean by covariate adjusted. And then a perturbation comes in, and these cells move into a new state. And that's what we are trying to predict, and that's what a state transition does. What is important, what is unique here is that it turns out the cells knowing their neighborhood helps the model. So the fact that you're moving a population of the cells as opposed to one cell is actually quite informative. And you can see this in the attention heads of the model. So you can see here on the bottom right, different attention heads. Some of them are basically single cells, right? So every cell is attending to itself, pretty much. Some of them are actually like pseudobulks, like its cell is like attending to just its neighborhoods, to everything else. And some are just kind of a mess, right? So it's basically integrating information. Every cell is integrating information about other cells in its environment to then better predict where it's going to end up post-perturbation. So that's what is unique about the ST model, let's say transition model. So that's one part. Everything that I showed you earlier in ST can be at the gene expression level. So you can pass it the cells in the form of just gene expression values. But it turns out you can actually give the model, not the raw gene expressions themselves, but some numerical representation of them. That numerical representation can come from an embedding model like SCVI or any other single cell foundation model. We ourselves trained our own module called state embedding. And this model, we have specifically refined it to be maximally expressive for this purpose. So these two modules come together, ST and SE come together to form a state. So once you train a state, now how do we actually evaluate it? So the evaluation, going back to where I started, we want it to be this context adaptation or context generalization framework. So we have observations across the data set for many contexts, and now there are held out contexts for which we only show the model a fraction of the data, and we want to predict the rest of it. So this is what I mean by context generalization. So this is what our test set is. And we have also developed an evaluation benchmark called cell eval that gives you all kinds of metrics for about how well your model is doing in these types of prediction tasks. What comes out of it is not unlike anything that you would do with a perturb-seq experiment. So we have really tried it to make it very biologically oriented in some sense. So I'm going to show you a couple of the metrics here that the state predicts, and comparing it to other benchmarks and baselines. So for example, this is Pearson delta is one of the metrics. So basically the log-fold changes due to the perturbation, what is observed versus what is predicted. We have the three data sets, the Tahoe data set, the parse data set, and Replogle, which is a perturb-seq, genetic perturb-seq. So two chemical perturbations and one genetic. And then you can see the comparison between the state and each of the other baselines. Another, which we of course very much care about, is our ability to predict all the differentially expressed genes. Again, you can see that a state does better than other baseline models. So in Tahoe, for example, which is again a drug perturbation data set, we're predicting about half the genes that are differentially expressed, quite well. And then in the Pearson delta, we're also doing quite well. So in cases where there's a lot of data for a lot of contexts, like Tahoe, we actually get much, much better at being able to predict what happens in a new context that was not in the data. So to summarize, this is our current state-of-the-art model. We are now working on other states. Hopefully you will see them. But a state one, again, it's an embedding model that gives you a maximally expressive embedding of every cell. These cells then go into this transformer architecture to then be transposed into a new part of the space, and then they get decoded into gene expression values. I want to end with the fact that, again, we know that this is a very vibrant kind of community that's coming around to building these kinds of models. We see a lot of papers. Everyone is the hero of their own story in their papers. So we needed to be in a place where we could actually see what is working and what isn't, what is working and what isn't. So we came together at ARC with help with us few sponsors, we actually built this challenge called Virtual Cell Challenge, where we specifically generate a data set every year for testing all of these different models that folks are developing at ARC and outside of ARC. So with that, I'm going to end. Big shout out to, again, a lot of people were involved in what I presented today, especially Johnny and Timmy, the Tahoe folks, and the entire Computational Tech Center at ARC, and just ARC in general. And I'm happy to take any questions.
Thank you for the wonderful talk. I have a quick question about the state embeddings. Has your team tried to transfer it to other downstream tasks in addition to perturbation response prediction? Because it sounds like to me it's a very useful embedding space you can learn. And I wonder what type of task would you think it's like interesting?
Yeah, so I think ultimately what tasks you focus on depends on like who you are and what you care about. So we, of course, have done a lot of work on perturbation, but in the paper, we also go through a number of other kind of classification, batch effect correction, and things like that you can take a look. I think the Tahoe folks also, in their paper, they did comparisons of state embedding to their model for a variety of other benchmarks as well. So there's quite a few tasks that people have looked at, like being able to predict gene essentiality, for example, from depth map, from the embeddings, and a number of other applications off the top of my head.
Thank you very much. I'll take a look.
Thank you very much for an excellent talk. I have two questions. Are you limited now by computer capacity to process big data or better data in a given biological context?
Very good question. I would say in this specific problem, which is virtual cell modeling, we are very clearly data limited. So there is this thing called the scaling laws where you can actually scale your model and see like how far there is room to go. We max out pretty quickly. It's just not a lot of data, and it's a lot of repetitive data. That's the thing about observational data. I mean, contrast that, say, for example, with DNA models that we have built at ARC, EVO series and EVO2 being the last of them. You have a 10 trillion training token data set there. So we are very much compute limited and going around asking for GPUs. Here is the exact opposite. Everyone was going around asking for data. And the more is the better. I think that's the answer.
And the second question is, given that there's emerging data in post-mortem patients and single cell data, analyzing that, how does your virtual cell communities work in that sort of system? Have you tried to model what happens, what the data looks like and should look like?
So good question. So we have this approach. We are testing this approach where, like, how can we look at everything as perturbation, right? So different cell types being perturbations of one another in some sense, age being a perturbation, disease being a perturbation, and how well will you be able to model these? So the answer is not well, right? So we are not doing really well with the current models that we have in kind of being able to access biology. And to be honest, once we do that, in some sense, we have built a virtual cell. I think that's the bottom line, but we're certainly not there.
We have an online question, which was also interesting to me, is what do you hypothesize? Is a hard question, but how do you hypothesize that we see that real challenge in one of those benchmark slides of the genetic perturbations being so challenging to improve on, whereas the chemical perturbations are somewhat easier?
I would say it has more to do with the just kind of a strength of the perturbation and the number of contexts in which they're seeing that perturbation. So, drugs, of course, make a lot of changes, and they're very strong. A lot of genes change, and you can capture that better. And we know this again because we have multiple doses, and we can see that at lower doses, where the changes are more nuanced, we do worse. But more importantly, I think what was game-changing about Tahoe is that you had like 50 contexts to look at, right? So for genetic perturbations, we just simply don't have that many contexts to be able to kind of generalize from the handful that we have to something that we haven't seen before. It's just very hard. I think, so it's this duality of like the strength of perturbations and the number of contexts in which we have observed that perturbation. So we need to solve kind of both of these problems. I think there's a question.
I have a question. Some of the cellular regulation happens at the subcellular or single organelle level. And that's something that's usually invisible to multiomics techniques. And I'm wondering, what's your intuition in terms of the state embeddings that you have? Do they end up also encoding differences in the subcellular regulation? Or would, for some context, data about that be needed?
Yeah, it's a good question. I think there is no, ultimately there is no like lossless compression, right? So what a state embedding does is like it takes your entire gene expression landscape, and again here we are talking about whatever a single-cell transcriptomic platform will be able to give you and summarize that in 2000s or so dimension, right? So what gets reflected in that, I think it's a hard question. So it depends. I think there are aspects of biology of organelles and everything that will be reflected in the transcriptome. And there are certain things that will be lost unless you actually emphasize that I want to be able to capture that. So you can act on the model to be able to retain its resolution for a specific kind of a specific applications that you have in mind. You say like, oh, don't forget this. Like these two are different. Just remember that these two are different. And the model will actually create embeddings for you that will be able to capture the part of the cell that you really care about. Otherwise, there is no guarantee that this compression would be lossless.
Thank you, great talk. Can you talk about the possibility using SE and ST-based model to predict multi-factor perturbations so as to develop a combination therapy?
Really good question. So there are two ways to think about kind of combinations. Of course, we didn't have any combination data. What you can do is like you can do it sequentially, so you can add one and then add another one. We actually have, again, in the state paper when it comes out, hopefully you'll see that Yusuf Seen has done some of this work as well. The reality is that where it's additive, you probably can. I think by definition, it's really hard to find synergistic and non-additive interactions. And I think we really need data to be able to model that. But the additive parts of this sequentially adding one and then adding next actually does decent in a lot of the tasks that I've looked at.
Thank you, Hani. And we will turn it over now to Manu Lonetti from CZI.
Thank you so much. It's really fun to talk right after Hani because I also am very invested in thinking about how to build a virtual cell model. And really what I think about virtual cell, I think about building a biological world model in very much the same sense that self-driving car companies are using world models to train their algorithms. Right? Today, most self-driving cars are trained by driving in a virtual simulation, not in the physical world. That is, of course, way more scalable than driving miles on the road. That also allows the self-driving cars to learn about corner cases of situations. What happens when a child darts right in front of the car in a safe manner. Waymo has a few examples also of what happens when a T-Rex crosses the road, and you should be ready for that. I think the potential in biology for creating a simulation of cells is enormous, right? We could test all the different drug molecules on a specific cell type, or we could ask how all different human mutations are affecting the body plan of a developing organism, for example. So the question becomes like, well, how do we get there? There's a lot of different aspects to this. When I think about it, I think about two things. I think about the fact that we're going to need to really define the basic input-output relationships of cells, and I'll come back to that later in the talk. But I also think that we're going to need to understand all of this in the context of specific mechanisms. First of all, because as biologists, this is what we care about. We want to understand things. But of course, to make all of these things actionable, for example, for therapy, we need to know what to act upon. We need to know how to control cells, even when we think about using cells as devices, for example. And so, to get there, I'm thinking about two different paths. The first one, related to mechanism, is really understanding the wiring diagram molecularly of a cell. Here is an example of this wiring diagram looking at proteins. I think we need to think about this across the central dogma, and Christina and Hani have given beautiful examples for this. But it's really about understanding and describing cells as an integrated ecosystem of molecules that interact with each other. And then the next question is, well, how does this specific wiring lead to cells being able to process, I'm sorry, I'm having the same problem that Hani was having, cells process information. And so I'm not going to say that I have any authoritative answers to both of these questions, but what I wanted to do today is show you how the work at the BioHub, how we're thinking about building technologies to really address these two questions in parallel. So far, a lot of the work that we've done is really focusing on the first part of the problem, which is really mapping the wiring diagram, and really, in this case, focusing on the protein wiring of cells. One of the key technologies that we've used for that is GFP tagging of individual proteins. Today, we can do that with CRISPR. We can do that at scale. We love GFP because, of course, it allows us to localize proteins using fluorescence microscopy. But as you probably know, GFP is also a fantastic purification tag. So we can use the exact same cell lines and do IP mass spec, for example, to measure protein-protein interaction networks. So originally, we really deployed this kind of assays at the level of individual proteins that became the foundation for a project called OpenCell, where we built one of the biggest, or the biggest, endogenously tagged GFP library in human cells. We've done that across more than 1,300 proteins. And for each protein, we can generate data that looks like this. This is 3D confocal microscopy in live cells. You can see nuclei here in blue, and the protein of interest, in this case, a Golgi protein, is in grayscale. And on the right-hand side here is a graph that represents the protein-protein interactions that we detect by mass spec. And a passion of ours is to build, intuitive, interactive websites. And you can explore this data online when you can, for example, click your way through this interaction graph, really jump from one protein to its interactor, and we hope start to get a sense for, again, the wiring diagram of the proteome. And this was a fantastic collaboration with many people, in particular, Matthias Mann for all mass spec. I really believe in the power of deconstructing the cell one protein at a time. I think we need more of these reference protein-resolved atlases. But of course, as you can imagine, there's like one big drawback, which is that these experiments take a long time to do. And if we want to look at cellular dynamics in particular and how cells get remodeled, well, that really forced us to rethink a little bit how to approach this problem and how to essentially reduce the dimensionality of the problem that we were addressing, right? I think this idea of how to best coarse-grain different cellular systems to be able to study them at scale is very general in our field, and I'd love to think about how that applies to the systems that you're all interested in. For us, what we decided to do in the first place is deconstruct the cell at the level of individual organelles, which makes a lot of sense in terms of eukaryotic cell biology, but it's just one way to think about solving this problem. So in our case, in this case, we're deconstructing the cell at the level of individual organelles rather than individual proteins. And these organelles become essentially biomarkers for cellular states. We can use the exact same experimental assay as I was presenting before. We're still using CRISPR to tag different organelle markers with GFP and with an epitope tag. We can use these libraries of cells and perturb cells and look under a microscope how cells are responding to these perturbations. But we can also do mass spec. And we can look at how the proteome of individual organelles is getting remodeled. And here what we're doing is using native lysis methods. So we're breaking cells open, but very gently using mechanical means. The cell is exposing its guts out, but everything is still intact. And we can, for example, isolate entire intact mitochondria and send them to mass spec and get the list of the thousands of proteins in that specific sample. This was a fantastic collaboration at BioHub, in particular with my colleague Josh Elias, who leads mass spec. So we did not invent this kind of mass spec assays. Actually, they were invented in this building, especially to look at lysosomes and mitochondria. But what Marco, Verena, and Frank, who were leading the experimental part of this work, were interested in was really to ask, well, how much would that concept generalize to different subcellular compartments, both membrane-bound, but also membraneless, things like stress granules or Peabodys, these so-called liquid organelles. You're going to have to read the paper to learn more details, but the answer was it generalizes beautifully. And surprisingly, it works extremely well also for these liquid organelles. So at the end of the day, they nominated 30 different protein markers that essentially cover all the different subcellular compartments that we could think about and deploy these assays. Today, we are pushing this into human stem cells, and we have a new library of GFP-tagged cell lines that is going to be distributed later this year to really start to generalize cell biology across different cell types, which is one of the big needs for our field right now. But to get started somewhere actionable, we were looking at HEC293T's transform cells for this first story. So the way to think about these experiments is that in each pull down, we're isolating one neighborhood of the cell. This is kind of like doing targeted fractionation, if you wish. So we're left with a data matrix where we're essentially identifying the vast majority, more than 90% of the proteins that we think are expressed in this cell type. These are the rows here, and each protein is going to be defined by its enrichment in these different fractions. And Duo and Kibom, who are leading the analysis of this work, spend a lot of time thinking about how to best represent this data. And at the end, they borrowed a playbook from single-cell RNA-seq, which was to transform this matrix into a graph. So this is one of these k-nearest neighbor graph. So in our case, we take each protein, we identify the 20 other proteins that share the most similar enrichment profile, and we link these proteins together in a graph, and we iterate. These neighbor graphs have a big advantage of denoising the data, because you're only looking at the neighbors. But also, graphs are very conducive to doing clustering analysis in a fully data-driven way. And in this case, for us, the clusters are going to correspond to different organelles, different subcellular compartments that we care to identify. This worked extremely well. And at the end, we can draw a subcellular spatial map of how proteins are organized in the cell. Here's a UMAP version of this. Each dot is a protein in the cell. These are color-coded for different subcellular compartments. You're going to have to take my word for it. But here, all this annotation is fully data-driven. Right? We're letting the clustering speak to itself. And in the paper, we show that we get the definition of organelles, which is actually better than what any other approaches could achieve. We also built a website where you can navigate all of this data. There are two things that I really love about this approach. The first one is that this data is inherently interpretable, but also includes a resolution of information that extends far beyond knowing which protein belongs to which organelle. Remember that underlying all of this is a graph, and here I'm showing you a part of this graph at the interface between, for example, the endosome and the Golgi complex. And here I color-coded different subunits of the same complex, and we're looking at complexes that actually work together in concert in cells. If you are not a cell biologist, It doesn't matter. But what I'm showing you here is that proteins that work together in the cell are actually next to each other in the graph, and that complexes that work together are also next to each other. Right? So there's a lot of interpretability that we get through this kind of coarse-grained information that is actually pretty high resolution. The second big advantage, and that's by design, is that these experiments are fairly easy to do. Right? So now we can look at cells before and after a specific perturbation. In our case, we started with a virus infection model. OC43 is a beta coronavirus in the exact same lineage as SARS, but we're all immune to it because we get that when we're kids, so we can do the experiments in the BSL-2 lab. And here, for the first time, we could really look at how the entire cell, again, each dot is a protein color-coded for different organelle, gets remodeled in a dynamic process. Here is a UMAP version of this. This is just for visualization purposes. Again, we can capture how the entire system gets rewired, but for any quantification, we're not working in two-dimensional space. We use much higher dimensionality of data. What was really interesting in analyzing this, and this is probably the most important take-home message from this part of the talk, and I think this is related to the question that was asked to Annie just at the very end here, is that when we compare the list of proteins that change in terms of subcellular localization, which the list of proteins that change in terms of their abundance, we did that with mass spec proteomics, we also did that with RNA-seq, there's actually very little overlap. And we don't think that's an artifact. We think that this is showing us that cells use these two modes of regulation, changing the abundance of things or moving things around, rewiring things molecularly, completely orthogonally. And as we look at the pathways that are enriched in these different groups of proteins, we get very complementary insights about what's going on in these cells. For example, in terms of downregulation, this is mostly related to an innate immune response, and this makes complete sense. This is a viral infection model. But in terms of subcellular remodeling, we're really being able to see a lot of homeostatic mechanisms. And in the paper, we show that we learned some fundamentally new things, for example, between the link between ferroptosis and viral infection in this model. So we're very excited about this kind of assays. We think that they're a right solution for deploying at scale assays that allow us to really understand how cells are wired. Max in the lab is building an entire data ecosystem to make this kind of experiments and the data itself, quote unquote, AI ready. So that means how can we harmonize all the code base? How can we centralize the data? He's building a data warehouse where he's scraping all of these kind of studies from the entire literature. He's building a community of collaborators around him, all contributing to the same code base. We're trying to get linked to the SC-verse universe. So single-cell genomics is really showing us the way of how to work together. And he's building web portals with chatbots so that all of us and students can really start asking questions on the basis of this data. We're hoping to have a preprint ready in the next couple months, but all the code is already public on GitHub. If you're interested, have a look, and please don't hesitate to send us an email if you want to talk more. Okay. So I've showed you how, our approach is to think about how to map the wiring diagram of the cell. This is not enough. We need to do this, again, across the central dogma. But now I want to switch to thinking about how we're going to understand cells as the basic unit of biological computation. Really what we want to be able to do is understand the molecular logic that links a specific input to specific output. But I think the opportunity is that we know how to ask these kind of problems, right? We can systematically perturb the system and systematically observe how it's responding to that. And at the end, it becomes more or less an AI problem to decipher this logic. I think what is very special in the case of cells is that they are nonlinear dynamic systems. And so to really map the input-output relationship, we need to know the starting state of this system. In other words, we need to be able to ask this question in a completely dynamic manner. So that really led us to think about using dynamic data, and especially imaging, which I think is one of the most natural modality to ask about cellular dynamics in the context of this question. What is missing in imaging is interpretability, right? But imagine if we could solve this. Imagine if I could take a picture of cells. Here's a bunch of cells here we're labeling actin, right? And I could interpret this picture in terms of a very specific molecular vector that describes the state, the wiring state of this cell. Now, I could follow the cell over time and really track how cells make decisions as they integrate information about their environment, explain the mechanisms behind it, and really start to learn how to predict cellular behaviors and control them. Okay. So how do we achieve that? Well, first of all, I think that this is a solvable problem because fundamentally, all the different molecular layers that we care about interpreting, be it transcriptomes, proteomes, metabolomes, are things that we can also read under a microscope. In other words, we can get the training data to do the interpretation that I'm calling for, and I think it's just a matter of time before we learn how to project all of this molecular information into something that we can read in live cells. The way we're thinking about this, then, is to build an AI interpreter from images to something that looks like an omics profile, right? So can we take a bunch of pictures of cells under different conditions and really use in situ omics methods to learn the ground truth of these kind of like molecular embeddings? To do that and learn generalizable rules, just as Hani was telling us about, we really want to look at cells in different corners of their cell state landscape. And we think that, for example, CRISPR screens are a very, very efficient way of diversifying cellular states in the context of these experiments. And here, I just want to give a shout out to a bunch of people working on this block that were huge inspiration for us, Paul Blaney, Ian Cheeseman, and Jonathan Weissman, through all of their work that really laid out the groundwork of how we're thinking about these experiments. So the search engine for these experiments are image-based CRISPR screens. This is what Paul Blaney calls optical pool screens. And a team at the BioHub has really been developing ways to deploy these screens very, very fast. We're building automation for different parts of the workflow, in particular in situ sequencing. We're focusing on small gene libraries that are just there to diversify cellular states and really go through large libraries of protein markers and pairing everything, in this case first, with classical Perturb-seq like single-cell RNA-seq. We're looking at, so far, 42 different live cell markers, which include things like organelles, but also things like biosensors. I think there's, for example, a caspase sensor somewhere here, autophagy sensor, et cetera, to really learn where is biological information encoded in image space. In other words, when we image, for example, mitochondria, which part of cell biology are we illuminating? If we do that in an iterative manner, we can really learn how to optimize what to look at under a microscope to ask the questions that we care about understanding. Also, in all of our assays, we are pairing fluorescence with label-free imaging. This is, in this case, reconstructing phase from bright field. And here are a few different examples where you can see that phase gives us so much. We can see mitochondria. So here are cells. I just color-coded here the nuclei to give some context. You can see mitochondria. You can see vesicles. In some of these knockout cells, we see weird vacuoles getting formed, and these vacuoles are always at the interface with mitochondria. There's really a richness in cell biology here that we cannot wait to explore. And so at the end, what we want to do is really identify a minimal set of markers. This could be label-free, fluorescence, biosensors, that would really allow us to interpret the molecular state of cells. But as a biologist, what I care about is function, right? Is this cell doing glycolysis or oxidative phosphorylation? Is it engaged into an autophagy response? Which phase of the cell cycle is it at? These are the kind of questions that we really want to be able to answer. Okay. So the rest of the vision then is that if we can make imaging interpretable, then we could build foundation models for cellular trajectories whereby we're starting with very vast amount of dynamic image data and really start training models that predict the future state of a cell, right? In the same way that language LLMs are predicting the next word in a sentence, in this case, can we predict the next frame in a movie? And you can imagine that if we can do that across a variety of perturbations, we can really start to learn generalized rules that sustain cellular behaviors so that when we're presented with a new trajectory, for example, a disease, we will know how to intervene and correct for it. The main limiting step to all of this is the speed at which our microscopes can operate. How can we take movies across millions of cells in parallel? And I'm going to go kind of fast because I'm out of time. This is the fantastic team working that I'm grateful to work with. We think that the imaging modalities to solve all of this is single objective light sheet imaging. With these microscopes, and my colleague Andy is one of the pioneers of this technology at BioHub, we're illuminating cells with a sheet of light that comes from the bottom at an angle. And you can imagine that in that configuration, when we move the sheet in XY, essentially what we're doing is we're doing something that's akin to Z-slicing through our sample, again, because there is that angle. Reto Fiolka at UT Southwestern then had this idea that, well, if we start moving the sheet, and here in this movie, we're seeing the sheet move faster and faster as you're exposing on the camera just a single exposure, at some point, if you move the sheet fast enough, what you're getting the entire volume of your sample just projected in one image. And by changing here the angle of the sheet, you can start to see here the entire 3D aspect of your cells. By doing this, we're building this microscope. Actually, we finished building it a couple weeks ago. Now we have to build the software to drive all of this. What we can achieve is a complete change of paradigm in how fast microscopes can operate. So 2 million cells per day, I would argue this is kind of what we can do with our fully automated confocal microscope. With what I just showed you, we can increase this about 500 times. And as you can imagine, then that really unlocks the potential for perturbation space that is going to be really amazing. And then fundamentally, what we want to do with all of this is then being able to map all the different trajectories, all the different endpoints that a starting state can access, and really all the bifurcation points that identify how cells make decisions about essentially their future. So what I'm showing you here is a vision. There's one last part to that vision, which is that what I think fundamentally we're doing is we're building cellular telescopes, new hardware platforms that are going to be useful for really a variety of applications. Of course, we want to put these platforms in an entire pipeline where everything is open, the hardware, the data, the models at the end. But in the same way that astronomy operates, I think there is a real opportunity for all of us to start working together. If I'm building really unique microscopes that could be useful for all of you, how can we build a system where you could bring your samples to this microscope and we could do experiments together? And this kind of, again, the astronomy model for cell biology is something that I would love to discuss and I think is very, very relevant for thinking about the impact of AI in our field. I'm going to leave you on this. Let me give a last shout out to all my wonderful colleagues and I'll be happy to take some questions. Thank you.
Any questions? Okay, I have a question. So I guess one of the slides, when you zoom in on a cell and you see kind of this readout of how it's evolving over time, how do we unlock that interpretability, especially when you show the scale of data that we can produce? I think maybe in single cell sequencing, this concept of cell state as defined by transcriptomics is somewhat accepted and has its flaws, but how do we extend that also to imaging data?
Yeah, that's a great question. Well, I think the most actionable way to do that is, again, to be able to translate images into the kind of classical omics representations that we all know and know how to understand, right? I think it is absolutely doable, and a lot of people are working on it, to start interpreting an image in terms of a transcriptome, for example. And I think that is probably the most immediate way to answer the question that you're asking. Then there is a much broader question that you're asking, which is also like, how do we define, the probabilistic envelope of all the different states that a cell can access? And here, I think what we, especially in cell biology, have to start embracing is the idea of just thinking about statistical distribution of things, not just trying to bin individual states in isolation from each other. And again, I think the single-cell genomics field is really paving the way for us to think about these problems.
I'll ask first. I really like the way you frame the virtual cell modeling problem as the car and world simulation. And I wonder if we treat the car or treat the cell as a car, to what extent do you think we need to perturb it to study its mechanism? For example, if we have a really good game now we are playing, we don't really need to have everything in that virtual car, right? We don't need to build an engine for that car, but we know how to make it move forward. So similarly, where do you think we are at now in terms of deciphering that car's mechanism to simulate it successfully?
So you're also asking, okay, to which degree do we need to open up the black box?
Yes.
To which degree is it okay if it's still a black box?
Yeah, this is a more general question.
As a cell biologist, my training is in protein structure. Well, first of all, because that's the fun part, right? Understanding mechanisms. But also, I think fundamentally, what we want to be able to do is intervene on the system, right? Design a drug, or actually more and more in terms of therapy, cells themselves are going to be used as devices. If we want to be able to engineer cells for them to be able to do something very specific, whether it's a CAR-T, whether it's something else, I'm going to argue that we need to understand mechanisms explicitly, because that's going to make all of our design, therapeutic design, way more specific and way more powerful. But that's an interesting debate.
Thank you.
We have a question from online, which is, I will try to represent it to the best of my ability. So modifying one gene at a time is a way of kind of understanding the ranges of states. But how do you calculate for, I guess, both the complexity and the fact that, as Hani's talk was earlier alluding to, that these are cells in a dish, and the complexity of the state that a cell can have also changes drastically in an organism.
Right, it's a super important question, and the question that's also I think Hani had a good answer to that, right? The question is, how do we build generalizability from? I think we need to design. These are probably the first experiments that we're all going to start to scale. Experiments that are addressing this specifically. For example, in perturbation data, how can you predict from single perturbation or small combination of perturbations the effect of much broader combinatorial space? Well, that's something that we can start to do with at least a limited number of data points. Let's prioritize these kind of assays. ARC had a beautiful example of that using the effect of mutational epistasis for protein function. And I think that's, there are many different ways where we can generalize that. The question of how do we go from cells to, tissues and organisms is also super important. We can deploy, one of the superpower of imaging is that it's deployable across different kind of biological systems, right? Some of my colleagues are starting to do the exact same experiments in a zebrafish, or actually, there are other fishes that are even tinier where you can see the entire organism, in an experiment at all different time points, right? I think also when you think about how fast stem cell research, differentiation, our ability to recreate something that starts to look like a human tissue, in addition, a context where we can still completely control and engineer its genome, for example, is moving so fast that I think there's going to be a ton of opportunities. And that's the route that we're taking to really start increasing biological complexity in these assays. time for one last question.
Yeah, so great talk. I had a question about...
Can you...
Sorry, yeah. So I had a question about the dark proteome. So I know there's a lot of proteins in which the structure A has not been solved, or more importantly, in your case, like understanding what protein function has on the cell. So how do you plan to incorporate, or do you plan to incorporate information from the dark proteome to inform your models to build a more holistic representation of the cell?
Yeah. Super good question. So there was a lot of talk quite a few years back about the dark proteome when systems biology was starting. The fact of the matter is, because it used to be like figure 5 of every paper, it's like, oh, I'm going to de-orphan a new protein. It's starting to be more and more difficult because we collectively have done a really amazing job at actually defining for human proteins the function of the vast majority of them, actually, with some kind of resolution. I think annotations are a mess because the same protein is going to have many different names, or sometimes it was studied in mouse models, but not in human. But I mean, frankly, it's the same thing. I think that dark proteomes really exist in prokaryotes, for example, and there's a real a gold mine to tap into. That being said, your question is still extremely relevant. I think that I'm amazed by how fast protein language models, right, are starting to apply beyond just predicting structure, but also predicting function. And I've seen on a few recent conferences people that essentially are opening up the latent space of the protein models and really start to say something very specific about the function, mostly like molecular function, but is this protein an enzyme? What kind of enzyme is it? What kind of, if it's a kinase, like what is its target distribution? There are more and more of these questions that can be essentially inferred just by looking at protein sequences, and I think that's an answer to annotating the dark proteome that could scale.
I think we had one more question in the middle, perhaps.
So I was wondering, with respect to non-coding variants, like which do not code for proteins, with respect to imaging, what are your thoughts on that? As well as, like I saw in one of your slides, you have like 42 different organelles or like type of things which you can tag. So I was wondering, what is the limit which you plan to eventually tag or think about, and like things about like correlations and overfitting?
Yeah. And I think that's also linked to the previous question, which is it's not been more than just the dark protein, but it's the dark, moleculome of cells and all the things that affect, for example, protein structure that are not protein, for example. We've done previous work that really showed that RNA molecules probably play a very essential, especially non-coding RNAs, role in templating protein-protein interactions. So I love your question. Imaging can read out pretty much anything that we care about, right? There are many different ways to tag, quote unquote, RNAs with MS2 loops, with different things. Bob Singer has done beautiful work and started an entire field around this to be able to do imaging on RNA molecules in the same way that we can do imaging on proteins. In fig cells, of course, then we can target any kind of RNA using essentially fish experiments, in situ hybridization experiments, in ways that are more and more scalable. So I think that also offers a window into what you're talking about. On the perturbation side also, we start having a lot of different tools, a lot of CRISPR modalities that target RNA specifically. And so both in terms of perturbing the non-coding RNAs and reading out their function, there are a lot of options that we want to capitalize on.
All right, thank you for all those great questions. We'll take a 10-minute break and be back at 4 for a few more talks and our discussion segment. Thank you. thank you for coming back. We'll continue with our program with Yuna from MIT. Thank you.
Thanks for having me. Thanks for the invitation. And I'm an assistant professor in biology and ECS at MIT. I'm also chief scientist at a scientific nonprofit called Tata Bio. Let me just see how this works. Okay, cool. So I'm going to shake things up a little bit, and then we're going to come out of the cell biology and human cells into something that is quite unknown, which is the microbial genomes. So I work in the space of functional prediction from sequences, specifically looking at microbial genomes. When we really consider the full diversity of life on Earth, it's really estimated that less than 1% of known gene sequences have validated functions. And we call this dark proteome or functional dark matter. We know these sequences exist because we have the sequences, but we don't really know how to annotate or how to interpret these genes. And even for really well-studied organisms such as E. coli and Mycobacterium tuberculosis, it's estimated that 1/3 and 1/2 of their proteomes are uncharacterized to date. And the truth is, we've only really scratched the surface of what we don't know. With environmental sampling and metagenomic sequencing, we're able to take samples from the deep ocean to soil and sequence the bulk DNA. And when we look at that, we realize that as more samples become available, the protein sequence cluster number increases linearly when we're looking at reference genomes, things that we can culture, but exponentially and without reaching a plateau for metagenomes. So what that means is that with this deep sequencing, we're just uncovering incredible diversity of sequences and biological functions, but we really don't know what to do with this data. So in our databases, we basically have billions and billions of protein sequences that have been sequenced, that are abundantly present across different microbiomes, but we really don't know what to do with this information. And microbial genomes are interesting to look at because when we think back to some of the most impactful discoveries in biology, CRISPR-Cas, restriction enzymes, Taq polymerases, all the way to antibiotics, all of these have microbial origin. So these microbial genomes, especially those that are uncultivated and potentially not possible even to cultivate, may harbor this immense potential for discovering novel functions that may have translational applications and significance. So my group works on this problem of, can we train a machine learning model that maps sequences to function? And of course, we need to first define what this function is. And depending on who you talk to, function is very different. So the way my group defines function is across these 3 axes. So one is just the natural language descriptor or annotation. So there's a whole field that's sort of looking at how do we map sequences to something that we can understand. You can also define function using molecular interaction between proteins, between RNA protein, and we talked a little bit about that in our previous talks. You can also think about function through the lens of chemistry. So if you think about biology as coordinated chemistry, then a lot of these enzymes and biomolecules can be thought of as things that can do chemistry. So that is another axis that I think about a lot. Today, I'm going to talk about function as molecular interactions, specifically protein-protein interactions. So in today's talk, I'm going to introduce why genomic language modeling and genomic context is even relevant for protein-protein interaction prediction. And then I'm going to introduce our new model called Flash PPI that utilizes genomic context in order to do linear time PPI prediction using PPI prediction as a dense retrieval task. This paper just came out today on bioRxiv, and also we just deployed it. So I'm really excited to talk about this today. And then thirdly, I'm going to talk about very briefly about what it means to deploy a model like this. And how do we make deployment of these models and decisions of the deployment such that we can make it more useful for biologists. A brief overview of genomic language models. So genomic language model is a large language model that is trained on the genomic corpus. Similar to LLMs that learn the semantics and syntax of natural language, genomic language models learn functions and regulations of different parts of the genome. So similar to how large language models might learn associations between words, you might be able to learn associations between genes or intergenic regions using a genomic language model. So the model that we're going to be talking about today is called GLM2. It's our multimodal and single residue resolution GLM. There are certain decisions that we made in building this model that I want to make note of. So it's trained on metagenomic context. So we wanted to really leverage the full diversity of sequences that are available to us. And we actually call the genes prior to tokenization. And then we tokenize the data such that protein coding genes are represented in amino acid space, and intergenic regions are represented in nucleic space. And the reason we did this was because we actually wanted to use this model for protein-protein interaction prediction. And of course, PPIs happen in amino acid space. So it made a lot more sense for us to represent the data this way. The way we train this model is using a mass language modeling objective. So we mask out certain percentage of the tokenized sequences, and then we ask the model to reconstruct the mass tokens given the context. And in doing so, what we're hoping the model will do is learn the relationships at the residue level between the protein coding genes as well as within the protein coding genes and intergenic regions. What's really neat about microbial genomes is that a lot of the components that interact tend to co-occur and localize in the genomes. And there is an evolutionary reason behind this. A lot of microbial genomes undergo a lot of recombination. And they have this operonic structure that's determining the transcription. So we want to make sure, the microbial genomes want to make sure that these genes actually occur next to each other so that you don't lose these genes. So we are actually utilizing this evolutionary signal because they're so co-located in the genome. So in this example, we have we have subunit A and subunit C. These are encoded in 2 separate protein coding genes. And depending on which lineage you look at, you see them pretty proximal to each other. So what we showed in our previous study was that GLM2 learns this co-evolutionary signal between protein A and protein C, or subunit A and subunit C. That corresponds to the dots here, that actually maps very close to the co-evolutionary signal that you can extract using a POTS model when you have a paired MSA. So these are evolutionary covariances between the residues that you can extract from multiple lineages of microbes in our database. Importantly, none of these models take in MSA. These are single sequence input. Of course, ESM2 is our baseline, doesn't learn the co-evolutionary signal because obviously it's only been trained on proteins, not protein complexes or genomic sequences. EVO is not trained on amino acids, so even though we're trying to account for this codon for each amino acid, we're not seeing any interaction between the subunits. But GLM2, because of its multimodal tokenization and the genomic context, we're able to recapitulate this co-evolutionary signal that you can see in a POTS model. And what's even neater is that this co-evolutionary signal actually does map almost directly to the PDB contacts. So things that are close in PDB space or in 3D space tend to co-evolve, and that's the signature that we're seeing. And that's this is the information that's been passed on through the tree of life, and we're sort of extracting this information. So we were really encouraged by this result, and we wanted to see how we can make this GLM2's latent understanding of PPIs even more salient. So we built Flash PPI, which is a system that, or model that does linear time prediction of proteome scale microbial protein interactions. So Flash PPI is built on top of GLM2. So we initialize Flash PPI with GLM2 weights. And then we fine-tune Flash PPI such that we're pushing protein-protein interactions, proteins that are interacting closer together in latent space. And also we're telling the model to predict the interface of the protein-protein interfaces. So that's actually really important because we have a joint optimization objective. So we have a protein-level contrastive learning that embeds proteins in a space that learns interactions. But we also ground it on residue-level contact prediction. So for each of these pairs, the model has to predict the contact between the proteins. So what this might look like is we have pairs of proteins that are interacting. These are interacting pairs, so we're predicting the positive contacts. And off that diagonal, we have non-interacting pairs, and we want to penalize that using contrastive learning loss. So what this joint optimization actually allows you to do is finding hard examples. So we do online hard negative mining. So as you can see, the model might predict some things off the diagonal to be interacting. But that is actually a false but difficult task that you need to teach the model that this is a challenging example, but you need to really not predict any contact. So this is actually a really neat way of integrating both the protein level information with the residue level information, and thereby make the model even more sensitive because you're able to do online hard negative mining without having the knowledge of which ones are hard negative or not. So the model is deciding on its own which ones are hard and which ones are not hard. So effectively what this means is that Flash PPI is learning a latent space of interactions, protein-protein interactions. So previously, latent spaces of protein language models or genomic language models might have been two proteins that are similar in structure, might be close together in latent space. But instead, what we're able to construct here is proteins that are interacting are close in this latent space, whereas things that are far apart are not interacting. And what that enables us to do is an inference that's very efficient. So when we have a proteome of a query proteome, we actually just retrieve the closest things that are in the latent space. And then we only construct contacts or predict contacts on those pairs. So we benchmarked Flash PPI on a held-out set, and these are all the sequence-only models. And what we show here is that Flash PPI can discriminate between a hard, challenging set between true and false interactions. And because of the joint optimization objective, the model is at the same time learning the contacts. So it also outputs the predicted contacts that actually maps pretty close to the ground truth contacts. And here we are looking at PPI interface prediction tools that are sequence only, that requires an MSA input. And in our case, is also sequence only, and we're able to reconstruct a lot of these interfaces that are ground truth. And we did ablate our model to see how our model architecture decisions and training decisions affected this performance. What we see here is that both joint optimization, meaning teaching the model not which proteins are interacting, but how those interactions are made, that is actually really critical to the performance of the model and generalization of the model on unseen protein pairs. And importantly, we included domain-domain interaction because the protein-protein interaction structures that we find in the PDB database are pretty sparse and limited. So adding the domain-domain interaction that probably learns similar patterns of protein-level interaction really helps diversify the data set and also generalize beyond what's been trained on. And finally, hard negative mining is also a very key important aspect of the model decision that adds to the performance. So we call this model Flash PPI because it's exceptionally fast. And we are able to achieve this because we're asking the problem in a different way. So previous methods when it comes to protein-protein interaction has asked this question of, given protein A and protein B, what is the likelihood of their interaction? So you get a score. So that means for a proteome of 5,000 proteins, which is a small, medium-sized microbe, you're going to have to make 12.5 million calculations. And even if you have a really fast method, this is just quadratic scaling of the prediction, which makes it prohibitive. Instead, with Flash PPI, what we're asking is, what are all proteins that A interacts with within the context, which is the genomic context, the proteome it's found in? So we already know that it's going to be interacting with things that are found in the same cell. So the proteome is the boundary. But now we're asking, what are all the proteins that it's interacting with in that proteome? So that means for a proteome of 5,000 proteins, only 5,000 searches are conducted. That means we can, empirically, that means we can do minute scale inference for bacterial genomes. I want to highlight that AlphaFold, if you were to do AlphaFold 3, which is the state-of-the-art model when it comes to molecular interactions, if we are to do all by all of E. coli proteome, that's going to take 100 GPU years. With our model, we can do that screening in less than 4 minutes. Yeah. OK, so for the next 5 minutes, I'm going to talk about what it means to deploy this model. I want to highlight some of the caveats of this model, because this sort of helps us determine how to deploy this model. So Flash PPI predicts microbial PPIs. So this is because GLM2 was trained on metagenomic sequences. So eukaryotic genes and genomes are actually out of distribution for GLM2. And it's also quite difficult to assume the same assumptions about eukaryotic genomes that we do with microbial genomes. Secondly, Flash PPI cannot easily distinguish between false positive paralog crosstalk. We were hoping it might be able to, but this turns out to be a very difficult task, even for AlphaFold 2 or AlphaFold 3 type of models. So in the case for diversifying protein families such as toxin-antitoxin or PE and PPE pairs, or fimbrial proteins and their chaperones, we're seeing a lot of sort of clusters of paralog crosstalk that we are not able to easily decouple. Thirdly, I think this is sort of a failure mode that we identified that's due to the model architecture, where distant homologs tend to be misclassified to be interacting. So I think it's because we have a single model that's projecting proteins into the same space. So even if the proteins that are remote homologs, they tend to be projected to a space that's kind of close in that interaction space. So we can mitigate this behavior by penalizing homolog interactions and also by removing homo-oligomer pairs during training, but we still see this as a failure mode in some cases. Some proteins somehow behave like hubs. They tend to be shorter proteins where they have more than 20 high-scoring interactions per protein. So this is something that we've also noticed. Some of these might be biologically true. Some of them might not be. So we need to think about how to serve this data to the user. So that means the inference pipeline is not just predicting which ones are interacting, but also doing filtering in order to give a proper and more likely to be real PPIs down the line. So given a E. coli K12 proteome of 4,400 genes or proteins, we encode all of these proteins individually. We make a vector database. We do top 100 retrieval per each protein in the proteome. And then we predict the contact. And this is the longest runtime in 200 seconds-ish. And then we apply a contact score filter. We identify hubs. We remove hubs that have more than 20 edges per node. And then we do a quick blast P between query target in order to remove the remote homologs or homolog relationships between the query and target. But it's also important to note that interpreting protein-protein network also involves understanding what these proteins do and whether these are worth going after in terms of scientific inquiry. So what we actually end up doing is we have an input genome, and then we call the genes to get the proteome. We predict the functional annotations within minutes because we are able to do embedding-based functional annotation. This is the work that was published earlier this year that uses context awareness to improve functional annotation or annotation transfer in prokaryote genes. And then using the same proteome, we predict the contacts between the proteins in the same proteome. And then we're able to look at both the genomic context, embeddings, embedding-based functional annotations, and the PPI network in a single view. So, so we built Flash PPI so that it can be integrated with our microbial genome interpretation platform called CCUP. So we have a FASTA input that is of a microbial genome. It can be incomplete. It can be a MAG. We predict the Flash PPI network. When you click into it, you can see which of these actually co-occur in the genome. Some of they might be a distal relationships. But also, this is a neat way to figure out which one of these are false positives due to paralog crosstalk. So, because tend to be is the case that, for instance, toxin and antitoxin pairs that are interacting tend to be close together in the genome. Importantly, we predict the interface directly. And then we can use the interface to highlight which regions are likely to be in contact within the sequence and the structure. So key takeaways from this work. So joint optimization at protein and residue levels improves both speed and sensitivity. So protein level allows you to get the linear time because you're projecting things into space where things are interacting or close in space. And online hard negative mining is important for challenging discriminatory tasks. Ooh. I also want to highlight that this speed and sensitivity is actually really important when we think about the full metagenomic space because we're considering billions and billions of proteins. So if you don't have high sensitivity nor high speed, then we cannot apply this technique to understanding metagenomic space. There's still a really large headroom when it comes to prediction performances. And one thing that we note here is that the residue level interaction and the knowledge of the contact interface is actually really important data that's needed for generalization. So what this means is that as we think about collecting future data, just knowing which proteins interact might not be sufficient if we're trying to generalize beyond what is a known set. So that sort of informs us, oh, given this model, we're able to generalize to a new set. But in order to improve this model, we actually do need contact information. And finally, as AI models in biology get more advanced, deployment matters. There's going to be a lot of caveats when we think about a lot of these models. And deployment that takes into account all the strengths and weaknesses of these models is really important. So the work I presented here today was actually done at Tata Bio, which is a scientific nonprofit that works in the space of genomic intelligence and understanding of this large databases of sequences. I just started my lab here at MIT, so I'm really excited to extend some of this work in my lab as well. Thank you.
Thank you, wonderful talk. So how about if we extend the genomic language model and the flash PPI to protein nucleic acid interactions?
The relationship between protein and DNA or protein?
DNA or RNA, yeah.
Yeah.
What are challenges?
Yeah, definitely a challenging problem. It's something that we're working on. Yeah, hopefully we'll have something to share soon. Yeah. Thank you.
Thank you.
Yes, coming.
Hi, so I noticed you filter for paralogs at the last step of your process. So I was wondering, why not filter at the first step for paralogs?
So that you don't have to calculate so many BLAST-P. So do you mean like at the beginning of a proteome interaction? Oh, I see, okay. So you kind of want to preserve all the proteins because maybe the ones that you actually care about might be in that paralog set. So you want to predict everything. And then at the very last step, in the end, I don't know if I can go all the way back. The paralog set is a known failure case, but it's only 12 out of this high confidence. So we want to limit the BLAST-P calculation between query and target because it is a computation that we want to minimize.
Yeah. So since this is a fast and kind of a static output, do you consider just run it through a lot of species or like data sets we have and publish them for the community to leverage? Because it's dumb. It doesn't matter to us how long it takes. Like we want a predicted outcome, right? With high confidence. And you can mark the parallel as caveats. And then it's good. Right.
Certainly. Yeah. So this platform that we're developing called CCub is basically a place where people can do this calculation for their species of interest, or we can do a bulk calculation. And this is an open community for people to both comment on, oh, is this like a real thing? Or I'm studying this, I have experimental validation for this. Or might sort of bring up new questions about potential interactions that have not been studied. Yeah.
Yeah, thank you. May I just quickly follow up? So, because these are static output, and how would I expect it to like vary given the change in future training data sets or like context switches, right? That's a very interesting question I have.
So in terms of the context switches, depending on your particular genomes, for instance, if you might have like a mycobacterium tuberculosis strain in your lab that you're working with, you can just run it on your own genome. So that's sort of a neat thing about it. So you don't have to rely on reference database because it only takes a couple of minutes to run. So that's one way to answer that question. In terms of model improvements, we plan to update the model with new PDB data and additional optimizations. So with that, we hope to be able to have version 2, version 3 of Flash PPI.
Thank you so much.
Thank you.
Any other questions? I had a question of, you have the language model annotation for some of these proteins, and then you have these contact PPI maps. I think that reduces maybe your search space a bit, but then how do you integrate over those to actually nominate something that you'd want to validate? Because you still have, the search space is still large there. So I was wondering, like, interacting both those functional annotations with
that's a great question. So this is still like a lot of information. If you know what you're looking for, this is a great resource. But if you're just sort of going at it, oh, this is an interesting genome, I want to just study what kind of new interaction might be present. We've actually integrated this with an AI LLM, like an LLM agent that integrates context, with PPI and how these genes occur across the tree of life. I don't know if I have this. Oh yeah, so given these two proteins, we're able to see that these two, we're able to do like a co-occurrence search across the database. So it's a lot of information. We recognize that, and we're trying to tie it together with a chat interface as well. Yeah.
Cool.
So on one of your slides, you emphasized, I think, like how you were able to leverage the operonic structure of the microbial genomes to kind of recapitulate these off-diagonal couplings in POTS models. And then you also compared with ESM2, which I think has a lot of microbial genomes in its training. Do you think, like, how much eukaryotic genomes kind of spoil the sensitivity to those
Yeah, I don't know if I can get all the way back on time. So basically, ESM is not able to learn the protein-protein interaction because ESM is a protein language model. So the input is a single protein. So it's never seen two proteins together. So understanding protein-protein interaction would be difficult. Yes.
Thank you.
Okay, cool. Thank you.
So one of the things Jean-Wei Li, who's of course on the faculty here, did starting when he was a postdoc in my lab was see this process of proportional synthesis where subunits of complexes were expressed at the same level. And in part, this, at least in E. coli, this was controlled by the Scheindel-Garnaut, basically tuned to make sure that they were matched. I was wondering if this is things that you could be learning in your genome model, because it's sort of exactly the type of thing that you would see.
Yeah.
You'd have that the proteins are things that interact and that the Scheindel-Garnauts were matched. And one of the really neat things that he showed was that when you had multi-subunit complexes where you had 10 subunits of one and one of another, like the F0, F1 ATPase, that the Scheindel-Garnauts were matched to match that. I'm wondering how much is that, can you see sort of attention being paid to Scheindel-Garnauts on top of, or is there an interpretability that you can dissect in there?
Yeah, this is actually something that we're working on with Jean-Wei, actually. So we're looking at how the intergenic region and also the the mRNA and the protein, the relationship, how the mRNA codon sequences as well as the UTR regions might be telling us more about the function of that protein. So yeah, I'm happy to talk a little bit more about it.
Very cool, but you're talking to the right person.
All right, thank you, Yuna. And now for our last talk, I'll turn it over to Ron from the Whitehead.
Wonderful, thank you. And this has been an absolutely wonderful afternoon of talks. And this talk will be a little bit different. It's mostly going to be perspective talk, maybe with some wild speculations. Just one data slide from our lab. Second will be a little bit different because I or my lab we are not developers of AI tools, but we're probably, like many people in the audience, experimental biologists trying to understand how life works and solve biological problems. And in that sense, to get to that goal, as you know, you kind of have to use the best tools available to get those questions answered. And these days, you can't ignore AI in that regard. So, broad perspective, AI right now is probably the biggest force at play on this planet, unquestionably. It's changing professions, businesses, and societies overall. And I would say the seismic shift in biology hasn't happened yet. So just using this tidal analogy here, I think as biologists, we're kind of sitting on the surfboard, looking out at the horizon, and we can see this big swell coming. And we don't know exactly how big it is, but we know it's coming, and we know we're going to have to ride that wave somehow. So I think that's where we are right now, and I think it's just a very exciting time, to see this intersection of what humans and machines can do to understand how life works. Is this the right thing? Huh? It is? This one? Okay, let's try that. Which button is it? Green arrow, that big thing in the middle. You see how technology limited I am. Okay. Well, here I thought maybe for the students and postdocs, I'd kind of provide some just perspective on how science is changing. The bottom line is whatever we're doing now is not what we're going to be doing in 10 years from now. And that's kind of, maybe the lesson of my lifetime in science, being maybe more senior than all the earlier speakers. But this talk will be about the importance of proteins in biology. And if you really, look back, well, this tells you how old I am, I suppose. The year I was born, the first protein structures came out. Actually straddled that 1958 to 1960 from Max Perutz. This is the first time we actually knew what a protein looked like. Okay, then I go on to graduate school. And this is really hard, I think, for probably students in the audience to imagine this, who now order genes. But cloning a gene was a thesis project at that time. And the nature of the genome was just largely unknown. I mean, there were a few hundred, maybe up to 1,000 genes that were cloned at the time. But what was in the genome, how many genes there were, it was just largely a black box. Of course, I could benefit from that because there were large superfamilies of proteins that were unknown, and I was lucky to be involved in discovering one of them, kinesin. But that just shows that there were just these massive unknowns about the players in biology at the time. Okay, these are kind of arbitrary events in my life. But anyway, that fit the year. But anyway, so come along, become now assistant professor, full professor. And around this stage is now the first time that the inventory of a genome was solved. First with yeast, and then, of course, moving on to humans. And information-wise, this is kind of the start of the World Wide Web and moving from getting knowledge from the library to knowledge through the computer. And this is really arbitrary time point, but about 10 years ago, of course, this single-cell RNA sequencing, which dominates this particular afternoon, so many people using it, first became available as a deployable tool. We have the use of CRISPR for genome editing. Okay, I moved to Genelia at the time. 2023, someone told me about ChatGPT. Really didn't know much about it. Now I'm using ChatGPT virtually every day. And the progress of what it can do in just a few years has been staggering. Probably the most rapidly, I would argue, LLMs are maybe the most rapidly developing technology ever made by humankind. And of course, AlphaFold solving this many decade-long problem in biology, almost a holy grail that the best minds try to solve, was solved of course, with all the databases for protein structure, but using an AI-based approach. Okay, now I'm at the Whitehead, but what is the world going to look like 10 years from now? Are arguably unknown. And this may be very scary to some who don't like change, but which is maybe all of us at some level. But at another level, I find this an absolutely exhilarating reason to be in science and in a scientific career, in the sense that our ability to solve questions that we're truly interested in is just getting better and better. The fact that we have to learn new things in our lives and in careers as things develops is also just a remarkably wonderful thing about this profession. But this is the reality of change and the unknowns of where we're going to be. So regarding this symposium, I thought I'd have first part like the here and now, like how AI is right now impacting the workflow of what it's like to be a graduate student or a postdoc in the lab. Just with maybe an example from my lab, and then I'll speculate a little bit of frontiers of what might be possible in the future from a perspective of a cell biologist such as myself. Okay, so the here and now, I mean, what are we trying to do? We're trying to get some insight into how living systems work. This comes a little bit few and far between in terms of a graduate thesis or a postdoc or even a career. And part of the reason for this is nature doesn't give up her secrets easily, and most of our experiments, the reality is, do not work. So our goal really is to make this coupling between designing better experiments that are going to give us important insight, to make that coupling as efficient as possible. And in that coupling process, we need to bring the best tools to the game to make this more efficient, because of course what gets us excited and puts joy in this whole profession is these insights that emerge. So of course, right now, as you've heard from today's session, there are a whole variety of AI tools available right here, right now, that provide also better insights and also allow us to design better experiments. So we're targeting experiments that are more likely to yield that kind of insight. And this is indeed happening right now. So I'll just give you, this is my one data slide, a couple examples from the last couple postdocs in the lab before Whitehead, who have now gone on to their own independent careers. This is Zhen Chen, now at Caltech. And what he was trying to do was to understand mammalian sperm, and particularly some of the unique mechanical properties of mammalian sperm compared to cilia and flagella from other organisms. And he did cryo-EM tomography, got structures, and found these colored things here, where there were some unique proteins that were not found in other kinds of cilia and flagella, something unique to sperm. The question was, what are these proteins? Because without knowing that, you can't do any further experiments. So there could be a traditional way of doing mass spec and getting this big, which we did, and then this big list of proteins that are found in the flagella, and then you look for candidates, but then you have to do a whole bunch of cross-linking experiments to settle and figure out which of those mass spec hits correspond to these proteins that you see in this visual EM tomography. That may have taken like 2 years to do. So Zhen decided to do another approach, which was to take the entire predicted structural database of all predicted folds of proteins from the mouse genome, and then create a computational pipeline that basically template matched each protein automatically to these protein structures in the sperm. And lo and behold, that worked. He could identify predicted proteins that matched these templates. So this was something that we ended up calling visual proteomics. point is, this allowed him to jumpstart and advance his project to what he really wanted to know, which is what these proteins are doing, and make mouse knockouts and do all of those additional experiments to really figure out what these proteins did. So more than a cool technique, it actually advanced his experimental work by years. Kristina Gladkova, now at Sloan Kettering, wanted to know how kinesin and dynein are regulated. So you switch one motor protein on, switch the other one off, or vice versa. So you get coordinated delivery of organelles to different parts of the cell. An unknown problem and a difficult problem to solve. Our initial experiments in this were not going in a really productive direction for producing insight until AlphaFold 2 came along. We got the predicted structure of one of the regulatory proteins, and that revealed a structural domain shown in this box. You don't need to know the details. It wasn't predicted by earlier structure prediction programs. And then AlphaFold didn't do this. It required a human being to look at that and say, aha, light bulb go off. This little unique structural element may be the regulatory structure that I've been looking for, and allowed her to do the targeted experiments to prove that was right. Again, kind of advancing what was possible as opposed to working in the dark and not knowing what this protein actually looked like. So it's really clear AI has really revolutionized how protein structure can be used to study biological problems right here and right now. And of course, not just in academics, but industry is exploding right now with this in terms of advancing drug discovery, designing and making new proteins. Companies are being formed, probably on a weekly basis right now. So things are happening in this space very rapidly. But let me now transition to the frontiers of what might be possible. So you already heard about this idea of a virtual cell from Hani's talk in CZI. It is an interesting question. Maybe we can go to the discussion of what is a virtual cell and what are the benchmarks of success. But it's just pointing in a general direction, I think, of trying to understand and advance our understanding of what cells do and how they work. And cells are, I think, the right playground for this, for the obvious reasons. And here's also a nice quote from Sidney Brenner. But cells are the fundamental unit of life. And if we don't understand how a cell works, we're not going to understand how an organism works. So it is the basic module that we really need to understand. And you already heard a lot of the questions that are being addressed by other speakers today here. For example, we heard from lots of talks to try to predict how cells would respond to perturbations, whether they're genetic or environmental. So effectively, these input-output relationships. Lots of work being done in this space. But I would say even a bigger goal is really just fundamentally understanding what a cell is and how it works, meaning it as an independent entity that is, can acquire and make energy, that is self-sustaining, that is replicating, and that effectively is an information processing device that controls what it does, what it moves, and all of its functions. And I would argue right now, we are at this stage where we know all the things that are in a cell, but we have the foggiest idea of the answer to this particular question right now. So this level of understanding how individual molecules in the cell create this emergent property of what a cell does is still very, very far from where we are today. But even go further, and this is Feynman's quote, if I, let's see if I can get the quote right. What I cannot create, I cannot understand. And therefore, you may even stretch this problem more. Could we actually create from component parts synthetic life? Now, of course, you're probably listening to that and thinking that is crazy talk right now. And it indeed is completely crazy talk. But look back 100 years and and talk to the people 100 years ago about doing precise genome editing to change the hereditary material and the phenotypes of animals or humans at a time when people didn't even know what the molecule of heredity was. But these are now things that are happening, obviously, right now. And therefore, who knows whether this may be possible in the future. So the question is, how close or far away are we from these questions? I would say right now we are very far away. And this is exemplified by even an interesting experiment done by Craig Venter's team to create a minimal life form containing the smallest number of genes possible. So this is a microbacterium. They were able to actually synthesize the entire genome of this organism, and then furthermore eliminate genes to identify a minimal gene set that allowed a replicating organism. That gene set has 475 genes. And this is heralded as a big success, but let me just say a very important thing, that this synthesized genome is completely lifeless. It has no lifelike properties, and it only comes to life when that genome is introduced into a container with cytoplasm, and that is what boots up the operating system, and we haven't the foggiest idea of how that works. Not only that, but when that minimal gene list was created, now this gets back to the dark genome, people looked at that list and said, well, we have no idea what a third of these genes are actually doing. So I think this is like kind of a statement that we are very far away from understanding what the simplest life form on Earth is, let alone that of a far more complicated cell. So the question is, how can we get to that understanding? And a lot of what we are collectively as a community investing time in to understand cells is the inventories, the transcriptomes, the proteomes, the lipidomes, the metabolomes, the organellomes, as you heard earlier. And right now, first of all, a challenge is how do we actually relate these different databases to create kind of a holistic understanding? But the other question is these inventories going to be enough? And do we need to add to that list of information? Do we need to understand information about the proteins in the cell? And I'm going to argue maybe a case that we do. But so we have inventories, we have copy numbers, et cetera, of the proteins in the cell now. We have at least some kind of idea of who's interacting with whom. But this is a giant achievement in science, but it also kind of belies some kind of ignorance that these are not, the information here is like a caricature of what are real machines carrying out all of this actual, what I would call animation of the cell. So each protein is an individual intricate machine, and that is actually doing the work to bring the cell to life. So for example, P10 is not a little diagram on this figure. It's not just a listing on an Excel spreadsheet. It is actually a three-dimensional machine that's carrying out its function. Now, the big challenge is proteins are individualistic and messy, meaning that you can treat RNA the same way, whether it's from a bacteria or a human cell or C. elegans, but each protein requires kind of, has its own individual behavior that needs to be figured out. And right now, this can occupy an entire PhD thesis to figure out just some aspect about what one protein does. This is kind of reminiscent of what I said, what in the 1980s, that it took an entire PhD to clone a gene. So the problem is, protein mechanism is very hard. And Also, I will also say that biochemistry now is a disappearing discipline because there's almost, the audience is rushing, you know, to what can be done with genomic techniques right now. So, you know, the real key questions are, can we understand, and what I mean by properties, I mean the structures of these proteins now available from AlphaFold, what these proteins are interacting with, but also the regulatory mechanisms, the activities, whether they're catalytic or other kinds of activities of proteins. Do we need to know the properties of these proteins encoded by the genome? And if so, what are the properties of these proteins that we need to know? So the protein structure at some level is now an accessible problem. But what about the other proteins, other properties that we need to know? How do we get them? And this is the critical thing. Can we speed up the process of understanding the properties and interactions of proteins? And will AI help us? So that we don't have to go graduate student by graduate student you know, over decades to solve this. My sense is that this is happening right now. So I'm not going to do a detailed journal club. I spend a little bit of time just pulling up some recent papers, and you also heard some more this afternoon. But I think we're in this stage right now where these properties of proteins are becoming accessible through AI tools, at least to an approximation. But that approximation may be incredibly useful for really collectively understanding how cells work. So, you know, first of all, I think it's not enough just to get the structure of a protein, because proteins are social, they work with other proteins. So you really need to know not just who's interacting with whom, but what that interaction looks like, because that really tells you about protein function. And there's this cool work coming out right now where mass spec data is being used to create a list with AlphaFold. Putting the two together, you now have maps of not like the arrows that I showed you, but actually what the protein structures look like of the interacting proteins. This is some work by Wade Harper's lab, who found a number of novel protein complexes using this mass spec with AlphaFold technique. We talked about this. Can one shine a light on the dark proteome? Again, there are papers coming out using AI that reveal kind of previously unknown proteins that belong to families or are related to other kinds of proteins. So we're rapidly getting new insight into that dark proteome as well. Critically, proteins are dynamically interacting with other proteins. And I would argue, you know, especially if AI is going to animate a cell and we're going to create a virtual cell, we're going to need to know at least an approximation of what those binding affinities and kinetics are. This is incredibly hard, even for biochemists to do right now. But I think there's some exciting work, including by right across the street, Regina Barzile's lab is doing really exciting work with this tool called Boltz-2. So this is also moving into the space where AI can provide information on that. Proteins are not static. They're moving. They have conformational changes. All of that is critical for their regulation. This was work previously in the domain of physics and physical models with MD simulations. There's a huge space right now where AI is working with MD simulations to create new models that vastly accelerate the accessibility of this conformational dynamic data. And you heard about this too earlier. Can we also learn about protein function, its localization, and activity from sequences? And you know, this features some work that you were voting on earlier, I think, the protein GPS model. But this is work from, again, Barzilli and Young's lab about predicting subcellular compartmentalization. This is another paper which had partial success, but also limitations on predicting very important things like catalytic rates of enzymes based upon protein sequences and data sets. Big picture, all of this is happening in the last 2 years. And there is just enormous work, progress that is happening right under our nose right now. So I think the scope of possibility of using AI to pull out this very complete, detailed information about what I would argue are the most important players that are animating life on Earth, which is proteins, and I will also add RNA for the RNA fans. But this kind of critical data is now, I think, over the next decade going to become accessible. And the question then is also, what do we do with it, to create this virtual cell? But it's very exciting times. So with that, I just want to say, beyond omics and inventories, knowing the details of protein machines will be important for understanding ultimately how life and cells work. AI tools exist right now that allow you just to do the good old-fashioned experimental work that we're doing. But it really can advance your thesis or postdoctoral work. And the last point is AI holds promise to open up what is currently a huge bottleneck right now of understanding details of proteins' function at scale. So with that, thank you very much. We can also skip the questions.
Yeah, we can skip the questions and we can move directly to our discussion segment. Thank you, Ron.
Thank you.
Missing. Christine. We're going to have just a few questions, and then we're also going to ask questions from the audience and also from the audience. So Matteo is going to check whether there are questions from the audience who are online. And I think I'll just start by just sort of asking each of you a little question, which was sort of what came up in my observations and what I was thinking. And it actually, it's interesting because I would say I'm more in the Ron's camp, right? Being an observer, but also really excited about the potential and always thinking, oh, we could use this, or we could use that, right? So being much more user than a creator in this field. And so it's really important to have the creators, but I think it's also really important that the creators and the users are talking to each other so that we can inform each other, right? What is actually our needs and what can you do, right? So this is I think where it's really, really exciting. And so Christina, I was sort of wondering, you're using these neuro-ODEs to track how individual cell genomes are organized over time. And I was wondering whether there are predictions of these models that surprised you, because so often when we find something surprising, is it sort of part of a discovery, that we discover something, we say like, that doesn't make sense, and then it bugs us. And I have had things which bug me for years, where I couldn't understand a result, and then all of a sudden you do another experiment. So I wonder whether the kind of science you're doing, if you get that same like, oh, that doesn't make sense, or this is a really surprising result.
So I will say, I did all this work with an expert in the Germinal Center, Ari Melnick and Darko Barisic. So I'm not sure. I'm not sure if there were expectations in terms of these. I mean, maybe it's all surprising. So they had this initial result that ARID1A and CTCF antagonize each other. And then when and there's a whole mechanistic story to that. And then when we did the modeling, we did these kind of rescue perturbations, we saw that ARID1A would rescue CTCF and CTCF would rescue ARID1A. So that gave us some confidence that, oh, like we're finding something that's been validated. And so I don't know. I think there's the potential to create unexpected results, things that you wouldn't go after until the model tells you, that there's sort of a rationale for this. But that in this particular project, I mean, the BCL6, I guess they did because it was predicted. Yeah.
That was predicted. But you had some others where you said you had to do mouse experiment, or you had to do some experiments actually to figure out what those meant. Because I also thought, oh, maybe those predictions are, so when you, and it's pretty wonderful to do actually the suppression of the phenotype. I really thought that was so cool. Yeah. But then many suppressors could also be sort of parallel kind of, I was thinking, oh, you're going to get a deubiquitinating protein, which sort of changes the turnover, and that's how you're actually suppressing, right? So I'm wondering how you can parse that out.
Right, and these models are statistical, right? So we're observing transcription factor activities and gene expression and modeling dynamics, but we're not really saying anything about how they're influencing each other, right? Yeah. So it's still less mechanistic maybe than we would like, but it's giving us
ideas, right?
a hand, like making predictions that at least some are validating.
So this is the feedback between theory and experiments. Hani, I think, you already said it, what is the virtual cell? Manuel may want to chime in with us too, but let's put you on, because we actually talked a little bit about this even last night at dinner, right? So what cell?
I mean, I would give a utilitarian answer maybe, because when I was a graduate student, we at length had discussions and arguments in the kind of computational community about what it meant to solve protein structure. Actually, for a long time, dynamics was a big component of it. Everyone was doing molecular dynamics as there are today, and I have friendships to date that we argued at length for hours what it meant. And when you look at CASP, which was a biennial challenge focusing on how models were progressing over time in this task, their metrics evolved. There were a lot of conversations and challenges on that front in early days. But if I take a step back and ask the audience of folks who are using AlphaFold, for example, how many of them do they even know what metrics were used in CASP to drive these progress? What matters in the end is that we reached a level of kind of accuracy that for experimentalists out there, they were comfortable inserting their protein sequences and get something out, and actually devote time and effort to going and validating experimentally those insights that came out of it. So along those lines, in genomics, for example, for the longest time, the way that we interrogate and understand biology is through knockout, knock-in perturbations followed by RNA-seq. I don't know how many in the audience have been doing this, but I know many labs that, anytime you come up with a new gene or a new insight about a gene, the first thing you do is knock it down and do an RNA-seq to figure out what happens, right? So if you can realistically replace this process with a model that gets even 80% accuracy of what experimentally comes out. I mean, if you do this experiment even between days, the accuracy won't be more than that. If you can replace this process that I just articulated with a model that does that, I mean, I would call that a virtual cell, but does it really matter? I don't think so. I think what matters ultimately is that people are using it and replacing something that's time-consuming and offer limiting, because you're doing one gene at a time and all of those things, and you can deploy it in silico and be able to do millions of perturbations in combinations in different contexts that might not even be accessible experimentally. I think that ultimately, to me, is not one manifestation of a virtual cell. But I agree that many people will have different answers depending on their applications that they have in mind. But ultimately, I think it's about that utilitarian view of these models.
Okay. I still don't understand what a virtual cell is. So the virtual cell is actually based on its disruption. That's what makes it. Because you have an immune cell which will have different disruptions will affect an immune cell than an epithelial gut cell.
I guess what I'm saying is that I think cells are also, like cell biology is also different things to different members of this community and the way they approach it, right? And they all have experimental processes for how they interrogate that cell biological system. Moving that process in silico makes it a virtual cell. It doesn't have to be a unified system that one model to rule them all. It can be different solutions for different types of processes. For us at ARC Institute, for example, it's everyone who does, again, like it looks at cell biology through this genomic lens of perturb-seq, knockdown, RNA-seq, and things like that. So for us, that manifestation of it is a model that does perturbation prediction. Mm-hmm. But of course, for many, it will be different. And for many in the audience, will be different. But we all understand that we can ultimately, with enough data and with enough sophisticated modeling, be able to move this process in silico. So in some sense, I don't think that virtual cell is ultimately going to be a singular model. Mm-hmm. But it's more, the first manifestation of it would be kind of in silico versions of like these processes that we do in the lab that are accurate enough to replace that process. So you don't have to do these experimental processes over and over again to interrogate different aspects of your cell biological system. Mm-hmm. So in the T cells, it will be different. In the context of differentiation, it will be different. In the context of development, it will be different. But the point is that we should be able to kind of model this in silico and get an accurate representation of that.
Okay, thanks. So Manuel, when you're looking at the spatial information and all the interactions, so one question that occurred to me was you are actually able to pinpoint like a few proteins which are identifying different organelles. Mm-hmm. And that you can use. So similar to what we're trying often for cell types, right? Where we're saying, okay, we can use this experiment, same approach, right? We can use that, what we've learned about, here is from all the single cell sequencing, and we can actually say, oh, these 5 are really what is identifying us. So I wonder, how well does that work cross species? Do you know?
Yeah, that's a good question. I mean, you're right. What we're trying to do is identify principal components, right, in a specific space. I think, organelles are just universal building blocks of eukaryotic life. So in this specific case, I think that they are universal principal components. I think there is part of the discussion.
It would be really nice, because then you could say, oh, you can make actually probes which go between species, exactly. Right? Because that is obviously with proteins the enormous. I mean, you had to tag every one of those proteins, right? You're not going to be able to do that in every.
I think what's interesting is to which degree. I mean, one of the big blind spots in cell biology right now, to which degree, some core mechanisms actually translate in different contexts, different cell types. As you're talking about different organisms, to which degree are we going to need to fine-tune what we're learning into each one of these contexts? I'm optimistic that we're not going to have to relearn everything across all the different cell types, because fundamentally, evolution has learned how to reuse the same modules over and over again. Is that fundamentally the rules that ground how these modules work together are being reused. And so I think that there's going to be a ton of generalizability that we're going to get because evolution has used that generalizability. So the way a mitochondria works is probably fine-tuned in different, in different cell types that have different metabolism.
Conditions.
That's right.
Or, a mitochondrium that's being transported, or a mitochondrium which sits at a synapse is quite different from a mitochondrium that sits next to the nucleus. So
I'm optimistic that we're going to get, at the general level, we're going to get like almost all the way there. And then we're just going to have to fine-tune with a few different measurements in different contexts to really have, for example, a cell type specific model that's going to apply to this specific cell type.
I was particularly excited about this label-free imaging, right? Because that would get us away from, making 20,000 insertions. So where do you think that can be going? Do you think that can go to really looking at cells in their communities? Could this go to organs or at least cellular communities so that you can figure out autonomous and non-autonomous effects?
Absolutely. And I think cell biology was born out of cytology, right? Just looking at the shape of cells in a tissue. To some extent, that's Ramon y Caral and all of these different approaches. And so, yeah, just the shape of biological objects, we know tells us a ton about what they do. We just don't know how to translate that. But I think there is tremendous potential in just doing label-free for everything and in learning how to interpret this information in mechanistic terms.
As far as I'm aware, Cajal, most of you know, right? Cajal drew all these cells of the nervous system and gave them names, and it turns out these are all different cell types which are now beautifully characterized by transcription, right? Which is actually part of, could be your argument, exactly. Right? Oh, I know this is an astrocyte because it has this. At the end of the day, we're finding the same list. We still have no idea how do you go from transcriptome to shape. So maybe we can. So that's sort of the interesting part, because here it's very clear shape and transcriptome is related. Absolutely no question, but we actually don't know. But anyways, I always find that absolutely fascinating. You're probably wondering, what's she gonna say next? I think this is because you're going to look at such a multitude of you are looking at such a multitude of proteins and such a diversity, so how do you decide what to actually follow up, or is that less of a concern for you? So is it like others? We should be doing that.
For the lab that I'm building as we speak.
Yeah. So what the graduate students doing?
So I'm really interested in biosynthetic pathways and how the microbes can do chemistry. So chemistry that we don't know how to do even. So I feel like that is an area that I want to go more into. How do we discover new types of chemistry that we didn't know about, and how do we utilize it and steer the design such that we can do new chemistry using microbial genomes or sequences. So that's the function that I'm choosing to go more into. But you can imagine, we're building tools, the foundation tools that can be used for other types of problems that people can go after.
Yeah, no, that's so. We were talking about how successful AlphaFold was, and we were talking about, and it's very clear AlphaFold was successful because of 2 points. One of you had it actually on your slides. One is evolutionary comparison, which has also, has been so successful for RNA structure. It's still the best thing out there, right? And the other was the existing maps, right? And for the evolutionary, most of you, except are focusing on humans, right? You don't think about other cells. I mean, you work on mice, okay. but anyway, it's pretty close. But so how, I mean, you're thinking about the universe. This is sort of more a question for everybody. Because often I feel like, this is like, many people are doing the same thing. Perturb-seq is obviously be done in many, many labs, and it's all this human cells, different cells, but there is all this life out there. So how can we use, you're giving us sort of an example maybe of how to use AI to kind of understand, really life, but also sort of to address some evolutionary questions. And so I was just kind of wanted to put that out as are there attempts, how are you using it?
There are attempts. I mean, in regulatory genomics, you need the data, right? And there are these kind of vertebrate genome projects that are generating chromatin accessibility in certain tissues for certain other organisms. And once you have that, it's clear that having more genomic sequence with function is going to improve the models. In the DNA language models only, these evolutionary ideas have been tried, but the sequence to function models that have been successful, it's just human and mouse so far, mostly.
I mean, I would add that I think human and mouse has been the bleeding edge of it because that's where most experimentalists have been generating data. But now that really for the first time as computational biologists, we live in an era that people ask us what kind of data we need for modeling, which is just like an amazing time to be alive, to be honest. And I think some of the answer that we have been given is exactly that regulatory networks, they have their own evolution, they have their own interactions, and following that in the evolutionary landscape would be quite useful for modeling the same way that protein sequences or DNA has been helpful. So there is also as more experimentalists, as these experiments become cheaper and more folks are generating data, we also are kind of trying to build a concerted effort to build actually data sets around other at least model organisms, if not just random organisms out there. But the reality is that because it requires this kind of omics measurements, and to be honest, again, if you go with morphology or even other mass spec-based approaches, or you can collect a lot of high-dimensional data from a lot of living beings. What we really know in the end is that we do need a lot of data, and we are nowhere near the kind of the scale that we need. So we know like something happens kind of magical around like a couple trillion training tokens in the models that we have trained, be it in like natural language or DNA or proteins. So that is kind of the where we want to get in kind of useful training tokens, and we're just nowhere near that when it comes to kind of biological omics data. So some of it is trying to have a concerted effort towards that. The other side of it is that in some sense that's why perturbation is so important, because it's in lieu of that. So we are kind of, instead of evolution messing with things and saying what works and what doesn't, we are the ones who are messing with things and measuring the consequences of that. But of course, you will need in the end a combination of the two to be able to solve this problem.
Yeah. What?
Just short answer. I mean, I think the potential for evolutionary data is just enormous. And the potential for AI to tap, this is just intuitive. Opportunity for AI to tap into that is, vast. For the simple reason that, yeah, we've done a bunch of experiments as biologists, but we have to be humbled by the fact that life has been doing experiments, for nearly 4 billion years. Almost every combination of everything has been experimented with, by life on Earth. And so if you think about evolution as experimental biology at huge scale, there's just so much information that is contained within that we don't know yet how to interpret or how to read. But you know, the potential for AI to learn things from the history of all of the experiments of 4 billion years on Earth, you know, we have, you know, records of that right now. And so, you know, it's more of a question of how to apply that information for problems that, you know, we're interested in. But I think it's super exciting.
Maybe that's the key to the real synthetic cell, making a cell.
Could be.
We should probably go a little bit to the audience, questions from the audience and online.
So Ron, you alluded earlier to how the cytoplasm is needed to boot up the synthetic genome. And I guess we've talked about proteins and RNA and DNA, but we haven't talked about all the other biomolecules. Does anyone have any thoughts on how AI can help us understand their role?
Well, let's give a quick question. I think that's a huge problem and one that we've kind of ignored in biology because we have easy techniques to measure macromolecules. We don't have really good tools, and we're just not in the habit of even collecting this information about all of the, you know, small molecules, everything from peptides to metabolites that are really, if you like, the buffer of the cell. And, you know, I think that's going to be enormously important. I'll just give you a brief thing. If you take Xenopus cytoplasm, and there's a way to make Xenopus cytoplasm effectively undiluted. And it does all these magical things, and lots of people study it. And you know, it makes spindles, does all kinds of properties. If you dilute it by 20%, all those functions go away. So there's something about all the small molecules and the concentrations of them that are critical for, you know, whatever the operating system of the cell. And just hardly anyone studies this problem. It's not an attractive problem to study. We don't have good tools for it. And there's not even a lot of attention to the problem, I would say.
Maybe, I mean, what gives me hope is that technologies are moving quickly, right? If you look at our ability to measure metabolites and lipids, for example, and you know, mass spectrometry is the leading kind of instrumentation for that, things are changing very, very rapidly in a very exciting way. Imaging mass spec is starting to, you know, to scale. Higher spatial resolution works beautifully for lipids because they're easier to ionize. And so that, because it's imaging, it also gives us like single cell resolved measurements. And so we're not looking enough at all of this, but I think the situation is gonna change very quickly. An amazing, you know, opportunity domain for all the trainees in the room that are looking for interesting questions to ask.
And there are, I think, like pockets of modeling happening. I think Josh Rabinowitz's team at Princeton, they have a generative metabolite model, for example, that actually used to generate things that looked like metabolites. And they looked for them in mass spec data, and they could actually find some. So there's like references are incomplete, to your point about like the expansion of those kinds of data modalities. I think the reality is that in the end, you know, like excitement brings more people into a field, and you know that brings in more resources, and it grows, right? That's, you know, like we look at AI now and all the excitement, but the reality is that it has gone through like 3 major winters. I mean, we have like experienced some of it ourselves throughout our careers. That there have been periods that the students will like just actively avoid a specific field of research that, you know, 5 years later would be like the greatest thing ever. And what changes is usually like some technological hurdle is overcome. You know, the reason that we have made so much progress on things like proteins and DNA comes down to the fact that next-gen sequencing really made it so accessible to collect all of this data. So I think as other technologies come online to look at other aspects of life, I think modeling them will also become easier.
Another Brenner quote, right? Is technology driving innovation.
So a big missing piece of information in cell biology is the so-called mesoscale. So once you get beyond sort of the size of structure of a protein and smaller than an organelle. And I'm wondering what's the potential for AI to really inform this? In particular, thinking that because we can now predict structures based on evolutionary couplings, et cetera, we can start our protein-protein interactions based on evolutionary couplings. We can now start to query protein-protein complexes or associations that are too weak to do by natural IPs. So that, because the difference between an IPable protein complex and a non-IPable might be a few kcals, which is a very, a few hydrogen bonds here or there. But then they just become blind because you IP and A doesn't pull down B, and how do you prove they interact? So I'm wondering, can we sort of tune our protein complexes to go from what Wade did with the, using AlphaFold multimer to finding all the things that have some signal evolutionary that they are binding to each other, but are too weak to be seen by standard methods?
Just have a first stab at that. I'm super excited about this area because, again, I think this is like a hugely ignored area of biology where there's kind of a gap in the tools that we have and the importance of the problem. And I think micromolar interactions are happening all the time in the cell and are incredibly important for function. Very, very hard to measure biochemically. I think if they're structure / AI tools, like I'm really curious what's happening with Boltz 2, like where the limits are on the affinity, but moving in that direction to bring micromolar interactions really in the fold of thinking about how biological systems work is going to be a really important problem in biology going forward.
I think it could also transform drug discovery because if you have something that's micromolar, the challenge to get a small molecule that pushes it to nanomolar is very small. So you might be able to get much smaller molecules and more specific ones to sort of glue proteins together.
I agree with you. We have this giant blind spot about how cells are organized at the 10 to 100 nanometer scale, right? For sure, AI has transformative capability there. The combinatorial space, though, right, is very, very large. Here again, technologies are changing very quickly. Cross-linking mass spec is really coming of age because now the new instruments have enough mass resolution to really map the cross-links. There are also new, actually, sample prep methods that make the experiments like 10 or 100 times faster. Cryo-electron tomography, and I mean, Ron had a beautiful example where they were looking at the sperm flagella and be able to identify a protein in situ, right? On paper, there's nothing. Physics tell us that we could do the exact same thing across all the different tomograms. There should be enough information in them if we can take these images with enough signal to noise, et cetera, to actually recognize individual proteins in these tomograms and, I think map the mesoscale organization that you're calling for. So again, that gives me hope because technologies are moving fast. Again, if something is not physically impossible, we know that some of us are going to have the ideas that would unlock this. My colleagues are working with folks at Berkeley, a way to increase, for example, the signal to noise in cryo-electron tomography 100-fold using like laser faceplates. Be happy to tell you more about this. But again, there's new hardware, there are new experimental modalities, there's new instrumentation that I think are closing the gap.
Probably should ask the last question.
Yeah, I have two online questions that I'll try and wrap into one and also close on, which is there's an argument about, or some of the worries perhaps around using certain classes of AI models is this concept or this worry of hallucination. Perhaps beyond that, how do we communicate the science that we do and the goals that we have in mind as to both, give validity to what we're doing, but also bring more people into the tent that Ron described, right? The idea that these tools are accessible and they can really accelerate what might take you 2 to 3 years into less time. And yeah, maybe that's a good question to close on, and I guess open to whoever.
Yeah, these tools are here to empower us, right? And I think to your point, another layer, not just beyond the tools, right? Also, what is immensely powerful is the collective intelligence. And so I think, if anything, together with the AI revolution, I think there's going to be a move in a way to work at the level of communities and consortia, because now that we have the tools to ask questions that are, at a broader scale than, the one protein at a time or one gene approaches that we're operating under, it's really going to take the collective to really think about this much higher level kind of mechanisms, for example.
I mean, I would say, I think, humans hallucinate all the time. We don't agree on anything. So I think we cannot hold these models to too high of a bar. In practice, again, you can recast the hallucination as a hypothesis, right? And you're gonna check that. Sometimes the information is out there for you to check, or sometimes you need to take extra steps, including experiments, to validate that. I think as long as we do know that that possibility exists, that the models can give you wrong information, I think that is probably sufficient for us to know how to deal with that. But at the same time, we are also getting better and better and actually querying the models about where they got the information from, and building citations into it and all that. But the more complex and the newer models, like biological models, actually make that harder to tap into the mechanisms. These models are just very complex, and interpreting the innards of them are really hard. There is this entire field of mechanistic interpretation that is dedicated to understanding what is it that these large language models actually learning, and how can we kind of peer into the model and understand something mechanistic about biology or language, depending on the underlying model. And I think those fields will also make incredible advances over the next few years as well.
So with that, I want to thank our wonderful speakers. Thank you so much. This was a really, really great I also want to thank Matteo, who is the spearhead of this. And our wonderful staff outside, who kind of made it all very seemingly. And finally, I want to sort of say something about the beauty of science, and how fortunate we are that we are in a community where we can have these discussions. And I have had so many ideas. Need to recruit a bunch of students. So that's really, really exciting. But also, in our audience here, there are people who have been supporting and have been really kind of wonderful partners of Whitehead. And I want to point out one particular person today. And so you probably all heard about Alison Liu. Who hasn't heard who Alison Liu is? Okay. So she just won the gold medal in figure skating. And the Olympic gold medal. And we have actually a gold medal figure skater among us here, and that's Tenley Albright, who won that 70 years ago, exactly this year. And then I should tell you, after she won the gold medal in figure skating, and she won also a silver medal in figure skating, I think, too, right? And many other medals, she became a surgeon and a scientist and a supporter of the Whitehead Institute. So it is just really wonderful because I think beauty, right? That's at the end, the beauty of cells, the beauty of movement. Thank you, and thanks everyone here.